Are you hearing me?
Yes, ma'am, you are audible.
Thank you. Dear guests, dear all presenters and the speakers, welcome to our international conference on the biological and medical science. I hope you will be very happy to participate in this event. I see Professor Dr. Hamdi Kemal. Welcome, Dr.
Yes,
Welcome.
yes, yes, welcome. Thank you, thank you. Hi from Turkey.
Sir.
Yes, okay, okay. Can we start? Okay or not?
Yes, sir, you can start.
Yes. Okay, okay, okay. Hi again. Hi from Turkey. Dear participant and dear organizing team. Firstly of all, I would like to welcome to participate. Also thank you very much to this organizing team. You are very valuable to me, really. So really, today I will explain important presentation because for us it is interested with our life microplastic, you know. If you don't take precautions, all things in the world will be badly affected, really. Um, maybe you know, I am a professor at Bozok University from Turkey, and I have 2 PhD, chemistry and pharmacology. Now, I am head of Department of Medical Pharmacology, and I am president of the Environmental Awareness Association. Of course, first time I have to explain plastic. You know, it is known as the first synthetic plastic badging produced in 1909. Polyethylene and polyvinyl chloride, originally made from polymers, were produced in the 1930s by BASF, the world's largest plastic manufacturers. Later, it started to produce polyethylene in 1933 and polypropylene and polyethylene in 1954, respectively. Industrial mass production of plastics began in the 1950. So maybe we can say something about plastic production too. Annual global plastic production currently exceeds 300 million tons. Really, please again listen. 300 million tons. Plastics make up the largest portion of marine debris. Thus, plastics are accepted as important pollution in marine and freshwater environments. It is known that the garbage produced in the cities is at least twice as much as garbage produced in the countryside, with the increase in production and consumption, and parallel with the economic growth in the countries the West are also increasing. So the increases in global demands for plastic in the world has become one of the 4 pillars of this plastic materials industry. Now, steel, wood, cement, and plastic with a strong growth, really. 20-25% of plastics such as electronic and natural media are in the durable consumer goods class. Most plastic products are extremely durable and can remain undissolved for up to 1,000 years. So really, plastic is present in every aspect of our daily life, in countless products we use every day, such as glasses, plastic bottles, forks, spoons, containers made of artificial foam, storage containers, toys, stationery products. So we can say this station to again, it contains substances such as bisphenol A, phthalates, azo dyes, heavy metals, and penetrates food and liquids at high temperatures, really. So microplastic, which were first defined as plastics smaller than 5 millimeters, defined by Thomson in 2004, continue to affects our lives every day. Microplastic can be divided into primary microplastic and secondary microplastic. For example, microballs used in cosmetics, personal care products, today's makeup, handcrafted and textile materials used in excessive amounts, plastic production pellets, or called primary microplastic, and in general the microbone in the cosmetics are made of polyethylene and composite materials in which they are in plastic in the sense. There is evidence that the primary microplastic source is synthetic fibers from clothes. Really, these informations are very important for our life. Secondary microplastic is exposed to mechanical, photolytic, and or chemical degradation, resulting in fragmented microplastic pieces and fibers, and occasionally broken down and formed by crumbling to less than 5 millimeters. Example of secondary microplastic can be said to be worn outdated particles, fibers that break from synthetic textiles, vehicle tire ratios, and various plastic particles. The source of microplastics are quite numerous and diverse. The first thing that comes to our minds is products such as shampoos, toothpaste, sun creams, face cleaning that we use as personal care and cosmetics. Later, microplastic may consist of such as fluids in special medical applications, oil and gas research, plastic industry, plastic containers used in homes, synthetic textile products, plastic waste, and microplastic can pass into our bodies from the water we eat, sea, and other food products we eat, skin contact, or respiratory tract. So, really, according to scientific article, microplastic was detected in the lungs, human blood, and feces, and plasmas or infants too. So really, when I explain, I am upset. Continue to. What are the advice of transition to people of microplastic? It can come from drinking water. One of the most common locations for microplastic is water, as most plastic waste accumulates in the seas and oceans. It can pass through purified water and cause a large amount of microparticle to mix with drinking water. Maybe in seafood too, it is fact that seafood contains microplastic. Again, microplastic will be formed due to plastic wastes spilled into the oceans, and the fish in the water will take them into their bodies and therefore people will be exposed to microplastic. Indoor dust, microplastic can be all over our home. Dust particles that accumulate on our containers, shelves, and other parts of the house also contain plastic. And dusting or rolling is one of the main ways of the human body. Food packaging, if you have plastic coated food or drinks in your home, it may contain microplastics too. In addition, canned foods, cardboard packaging, PET bottles are also likely to contain microplastics too. Dermal exposure, persons exposed to microplastic during washing, such as hand, face wash, water contaminated with water, or face, body scrubs containing microplastic too. So microplastic found in the air can also be taken by respiratory when the water containing microplastic evaporates. Some microplastic may enter the atmosphere. It can also be dispersed through the wind while a car is driving. Your tires can be mixed with slow wear and disintegration due to the friction and heat. Many pieces of clothing made of polyester contain small plastic components added during production. When you rub the surface of polyester clothes, thousands of small fibers can be released into the air. So I gave about microplastic and plastic too. So now I will explain studies on the damage caused by microplastic to living things. Really, for these academic areas, there are a lot of manuscripts really nowadays. But in the world, I think first time I studied this about microcyclic plastic and damage of plastic too. So this manuscripts is very important because Gaio and colleagues in 2021 was published in Frontiers in Marine Science, and they found that microplastics affects chromosomes in zebrafish. Really, where these manuscripts is very, very, very important because these microplastics affect chromosomes. I am sure in the future these microplastics affect our chromosomes too. So other studies, GSAMP, so state in their report in 2015 that the alternating engagement of microplastics can cause, please listen strongly, can cause change in chromosomes and lead to infertility, obesity, and cancer too, unfortunately. So other study, a study in scientific reports also state the microplastic can be found oceans, rivers, sediments, sewers, soil, and even table salt. Depending on the particle size, it has been observed that microplastics can accumulate in least 3 tissue in most subjects: liver, kidney, and intestine. In addition, the accumulation of macroplastic causes various effects on biochemical, biomarkers, and metabolic systems, compromising health or mammals. Really, really very important studies. Other manuscript, in vitro tests reveal the potential effects of macroplastic in cells on gene expression, ve cell viability and pro-inflammatory response to. So you can read all manuscript, so you can understand that we can do something to stop this use plastic. Other study, microplastic can affect metabolism by altering metabolic enzymes or distributing energy balance too. The metabolic effects of microplastic can have a negative impact on humans by increasing or decreasing energy expenditure, reduced food intake, and regulation metabolic enzymes. So in the world, people don't want to be anything, action, for example, sport like. In vitro studies show that macroplastic are concentrated in the gills, stomach, and metabolic crop systems and cause adverse cellular change in fish. And other study exposure to microplastic is also related to oxidative stress. Listen again, oxidative stress, cytotoxicity, and other tissues too. And macroplastic cause chronic discomfort, swelling, cell growth, and death, as well as immune cell dysfunction, unfortunately. Macroplastic can affect metabolism and change metabolic enzymes or disrupting energy balance. Macroplastic metabolic effects or by increasing or reducing energy consumption, reducing nutrition intake, and regulation metabolic enzymes can have a very negative impact on people, especially on people too. According to Prata 2018, if long-term exposure to macroplastic can cause inflammation and irritation, DNA damage, and cancer too. So DNA damage and cancer. Ferhat and his colleagues 2011 said that macroplastic can cause chronic damage in the cells, the production of immune modulators, and the incorrect stimulation of immune cells and autoimmune disorders. Antibodies against their own antigens will be produced as a result of this chain of events. So macroplastic can cause bone loss by stimulating osteoclasts when they reach the bone too. Please again read this manuscript. They reach the bone too. Ingested macroplastic can cause cellular toxicity to human live cells. Very small plastic particles can cross cell membranes, thus helping to increase to the bioavailability of plastic-derived toxicant too. So I would like to talk a little bit about our work on plastic bottles. We investigated whether plastic additive fast into food and drinking water that plastics come into contact with, and we found that a significant amount of polymer additives pass into the water we drink from plastic bottles every day. We used the LCMS isotope instrument to confirm the presence of these molecules. In our study, first of all, 500 milliliter plastic bottles, which can be easily purchased from the markets, were used. As a result of the analysis of this water in the LCMS isotope device, it was determined that 3 different additives passed from the plastic bottle to the water. One of them is Chimasorb 61, which is UV absorber, and the other 2 are stabilizers, oleamide and Irgavos 168. In the second stage, 500 milliliter petal bottle water was kept in the sun for 8 days with its mouth closed. The water passed through similar preliminary preparation stage was analyzed in the device, and it was determined that antioxidant 2246 and BHD substance passed into the water in addition to Chimasorb 81, Irgavos 168, and oleamide substance. So, when you drink a plastic bottle, we can drink water and 5 chemical compounds together too, unfortunately. And maybe, you know, in Turkey and some countries too, I give different conference and I join TV programs, radio programs too, because for this area I have studies and I have my books too. One books, Love Without Water Doesn't Live. Maybe its name is very different or very different. Yes, you are right. In this my book, I talked about water-related issues and tried to explain how drinking water in plastic water cause harm. And other my book, Low Plastic Kills, I embraced the pollution of plastic bags. I tried to scientifically explain how plastic bags harm nature and living things. Our other book, Suzu Learns to Protect Nature, which I am considering has just been published. We have written a storybook so that primary school students and kindergarten students can understand our academic work. So, as a result, it is really difficult to reasonably assess the 3 effects of microplastic on human health due to the lack of robust data measuring human exposed to microplastic, due to the increasing severity of microplastic pollution in both aquatic and terrestrial environments, the processes and mechanisms related to the entire of microplastic into the human body and their toxicological effects on human health need to be comprehensively investigated before long. So thank you for your participation and thank you very much for listening to me too. I think very fastly because my time is okay.
Thank you, Doctor. Thank you for your so interesting and helpful presentation. As we know, this topic is so actual in our area because plastic things every day we use at home, not just at home, in our
Yes.
environment. So because I will say the good damage for our uh, health.
Yes.
So
So we have
there
to,
is
yes, we have to use glasses instead of plastic in our life.
of course. Thank you very much again, dear guest. Have you any question for Professor Hamdi Temel? I see Matthew. Have you any question?
Yes,
We
this
can write, we
presentation
can
is
write your
about,
question also
yes,
in chat box too.
okay,
Okay, if
thank
you
you.
have any question, thank you again for your so helpful presentation.
Yes,
Okay,
thank
as a
you.
presenter, thank you. As our keynote speaker, Dr. Amara Diaz. Dr. Amara Diaz from Department of Life Sciences, Supervisor Ferit University of Engineering and the Information Technology, Pakistan.
Okay, starting from the main topic, anticorins, diterpenoids with anti-cancer potential. So first of all, I would like to tell you about the importance of natural products. Nature has always been an indispensable and underexplored source of therapeutic agents since the time immemorial. The number of first in-class drugs approved by FDA from the year 1999 to 2013 is 112, and out of these drugs, 28% drugs were based on national pharmacores. According to an estimate, 80% of the overall rural population of the world, as well as 75% population of the developing countries, still rely on plant-based products for curing certain diseases. In addition to that, more than one quarter of the prescribed drugs in developing countries are still dependent on plant-derived products for the treatment of various disorders. Now coming towards my main topic, that is anthocyanins. Anthocyanins belong to the diterpenoid class of natural products, which are highly abundant compounds present in the genus Isodon, Paris, Naphthalinium, Diplospora, and Croton with highly oxygenated and diversified structures. These plants are mainly found in China and possess a significant importance in traditional Chinese medicine due to a high spectrum of biological activities possessed by them, like anti-cancer and anti-tumor activities, like leukemia, gastric cancer, lung cancer, colon cancer, nasopharyngeal cancer, liver cancer, and cervical cancer, etc. Now, I would like to introduce some of the biological activities of anthocyanins. Antcorine diterpenoids have become the potential candidates for the development of anti-cancer products. Various in vitro and in vivo studies test their efficacy in terms of cytotoxicity, anti-inflammatory, immunomodulatory, and anti-cancer behavior. Here I mentioned the various activities of antcorines and the molecular mechanism working behind these activities in various kinds of cells. Anthocyanins like ponicidine, xylopic acid, flexicoline, ribudoside, oridonine, and some other like arioclaxin V are used as anti-tumor, anti-angiogenic, anti-proliferative, hydrophilic and lipophilic agents, anti-proliferative agents, natural sweeteners, anti-inflammatory as well as antioxidant agents. Here I am shown some of the biological activities of anthocyanins with their chemical structures and natural resources. Oridonine is mainly found in genus Isodon. Its chemical structure can be seen over here, and its biological activities like anti-cancer and anti-inflammatory activities have been explored in detail. Likewise, Areoclexin B is found in the genus Isodon in the plant species like Isodon, Areocalyx, or variety Lexiflora. Its chemical structure has also been shown over here. It is found in the dried leaves of Isodon Areocalyx, and its activities like anti-inflammatory, antibacterial, and anti-tumor activity have been explored in detail. Likewise, flexicolin A is also found in genus Isodon in the species Isodon rubescens. Its chemical structure has been shown over here. It is mainly found in the leaves of Isodon rubescens, and its anti-proliferative activity has been explored in detail. Here are other anthocyanins whose side act as a natural hydrophilic and lipophilic agent. Glycocollaxin A is found Dosia japonica, root, stem, and leaves, and its anti-cancer as well as antibacterial potential has been explored in detail. Ponicidine is also known to be a cytotoxic, anti-tumor, antioxidant, apoptotic inducer. Xylopic acid is found in the plant Xylopia ethiopia fruit, and its anti-proliferative activity has been explored in detail. The anti-cancer activity of these compounds have been explored in detail. Cancer, the second most fatal human disorder, has been marked by the deregulations in the epigenetics, molecular and metabolic processes inside human body. Natural products serve as the most effective sources to provide potential anti-cancer scaffolds to eradicate such human metabolic disorders. Cell cycle arrest is the main phenomena which causes the anti-cancer activity. Various anchorines have been reported to induce cell cycle arrest at G0 by GM phase, as well as G2 by M phase. The molecular mechanism which acts on the backside of the activity of these plants is known to be to affect the NF-kappa B pathway. NF-kappa B constitutes the transcription factor which is involved in the cellular cytoskeletal remodeling, survival, proliferation, adhesion, as well as apoptosis. NF-kappa B phosphorylation was known to be inhibited by the activity of carcasine an anchorin from the croton microns. The proposed inhibitory action was studied in human T lymphoma, Jurkard, E6.1 cells, and human acute promyelocytic leukemia in the cells HL60. IC50 values of kerosene acid were noted to be 4.2 micrometers in 4.2 micromolar in Jurkard cells and 4.36 micromolar in HL60 cells. Areoclexin B, another anchorin from Isodon areocalyx, is known to downregulate the activity of NF-kappa B and its downstream targets, that is COX-2 and inos, due to the presence of 2 alpha and beta unsaturated ketones in case of hepatocarcinoma cells. Likewise, it is also known to affect the AKT pathway, and who discovered the uridanine inhibited who discovered that uridanine inhibited the HeLa cell lines activity to proliferate in a dose and time dependent manner. Here are the mechanism of anti-cancer potential of various anchorins against different cell lines. Uridanine is known to affect the oral, gastric, liver, nasopharyngeal, colon, lung, bladder, leukemia, cervical, pancreatic, and esophageal cancers. The cell lines used for the study of these natural products were OSCC UM1, SOC25, BXPC3, MCF7, L929 cells. The molecular targets which were mainly focused were cyclin B1 that was down-regulated, cyclin D3 which was up-regulated, cyclin D1 that was up-regulated, PCDK1 was also upregulated. P21 was upregulated. Cyclin A2 was upregulated. PI3K by AKT was known to be inhibited. P53 was upregulated. P50 the phosphorylation of P53 was also known to be increased. Likewise, the level of BCL2 anti-apoptotic protein was known to be downregulated. Heat shock protein 90 was known to be downregulated. Likewise, the apoptotic inducer BAX was known to be upregulated. Likewise, CDK2 was known to be downregulated, and other apoptotic as well as anti-apoptotic factors were known to be modulated by the action of aridonine. Likewise, the molecular mechanism of some of the endcorine has been shown over here. Synergistic effects. Hesperidine, a flavonoid, has remarkable biological activity, but it is poorly available to its low water availability. But when combined with endcorines, its biological activities were known to be enhanced. It can be combined with riboside, which can enhance its anti-cancer activity and act more effectively in case of breast, colon, and prostate cancer cells. Conclusion. This research summarizes the status of endcorines as the newer and bioactive compounds against various human disorders, and more specifically focusing on their anti-cancer activity. The data presented in this article point out the molecular mechanism working behind the cytotoxic behavior of these diterminoids, indicating that these compounds exert anti-cancer effects on cancer cells on as low as 0.39 micromolar in case of HGC 27 cells. The synergistic effect shown by these compounds can be utilized to maximize behavior benefits from other drug candidates. It has also been suggested through various studies that NF-kappa B and AKT pathways were involved in exerting potential anti-cancer effects, while apoptosis was noticed to be induced by following raw scavenging and pherodopsis. Various studies confirm that these compounds may be further channelized as potential lead compounds against cancer and related metabolic disorders, but further rigorous investigations are highly invited in order to channelize these compounds in developing potential anti-cancer compounds. These are the references which have been used in this study. Thank you so much for joining and listening my presentation. Any query is highly welcomed.
Okay, thank you very much, Dr. Amara, for your presentation. Have you any questions, dear guests? You can write on chat box, and also you can raise your hand if you have any question. Okay, if you haven't any question, we can go to other presenter, to other speaker, Dr. Majida Rashid. Start from the presenters, Noor Arifa.
Okay, first and foremost, thank you, Chairperson. Good evening, everyone. My name is Noor Arifa Amira, and I'm from Kelantan, Malaysia. So currently, I'm doing my Master of Science, specifically in reconstructive sciences. So today, I would like to present our project entitled "The Analysis of FGF10." Copy number and expression in non-syndromic cleft lip and or palate among Malay patients. May I know
change your slide, please.
Wait. Okay. Is it okay?
Yes. Okay.
All right. The orofacial cleft is one of the common congenital defects among the newborn. So we could see most of them are isolated or non-syndromic. and some of them are present due to the syndromic conditions such as Kalman syndrome, Van der Waals syndrome, and Treacher-Collins syndrome, as well as the Pierre-Robin sequence. So for your information, the prevalence of orofacial cleft is diverged across countries and races. So the International Prenatal Database of Typical Oral Cleft has recorded overall prevalence 9.92 per 10,000 of births from 2000 to 2005 across 30 countries, includes United Kingdom, United States of America, Europe, Japan, Australia, and United Arab Emirates. Meanwhile, another study reported by Oliver, the rates ranging from 1 to 500 to 1 to 2,500 live human births in 2012. So in Malaysia, a study was carried out in 1990 involving the patients with cleft lip and palate, whereas the Malay ethnicity contributes the higher occurrence in CLP with 88.6% from 526 patients, followed by the Chinese with 8.7%, Indian with 8.2%, and the least is among other minor ethnics with 0.2%. As we can see, that cleft lip and or cleft palate occur during the early phase of the pregnancy from week 4 until week 9. So this is due to the partial or complete lack of fusion of mouth and nasal structure. The most well-known type of the cleft are the cleft lip, cleft palate, cleft lip, and cleft palate, which is with alveolar involvement. So for information, the alveolar involvement is the regions that hold the teeth at the anterior of the mouth. And then the conditions of the cleft could be classified as unilateral and bilateral condition. Basically, NSCLP involves the combination of genetic and environmental factors. Among the known environmental factors, the passive smoke exposure, viral infection, lack from folic acid intake, just to name a few. Thus, it will complicating the identification of causal genes. So in this study, we were investigating the fibroblast growth factor 10, which is also known as FGF10, to be one of the causal factor in the incidence of cleft lip and or palate among Malays. The previous studies have shown the association of the genes with the occurrence of NSCLP, but to date, there is still no study conducted on FGF10 in the Malay ethnicity yet. For information, the FGF10 is one of the 22 members of FGF family. So usually, the fibroblast growth factors will bind to their receptors, which is also known as the fibroblast growth factors receptor, that will initiate a wide range of biological responses, including the cellular proliferation, differentiation, migration, adhesion, and survival. And several members of FGF and FGFR families are expressed during the craniofacial development, including FGF8 and FGF10. So the role of FGF10 is to mediate the biological responses by activating the FGF receptor 2B, which is FGFR2B, with heparin sulfate in paracrine manner. So both are expressed in mesenchymal and epithelial tissues respectively. The previous studies have shown the variants in FGF10 may be involved in the risk of the cleft lip and palate. Okay. So hence, the aim of this study is to determine the potential correlation between the copy number of FGF10 and the mRNA expression in Malay individuals affected by the NSCLP. So the methodology that we use in this study is we collected the blood as our samples. 74 samples were collected from the consented participants, comprising 37 patients with NSCLP and healthy individuals as controls from Hospital University Science Malaysia from 2021 to 2023. So prior to the collection, health screening was carried out by the clinician to ensure the individuals involved fulfill the inclusion and exclusion criteria as patients and controls. And then after that, we withdraw the 3 ml of blood to extract the DNA and RNA from it. So after that, we determine the purity and the concentrations of the nucleic acids using the spectrophotometer before proceed with the downstream analysis. So the purity chosen were in the range of 1.8 to 2.0. Next, for the downstream analysis, we conducted two analyses. The first one is a copy number variation assay for the DNA and the mRNA expression for the for the RNA. The Tegman copy number assay was used to determine the copy number of FGF10 across the genome using real-time PCR method. So we diluted the samples to 5 nanogram per microliter. We used the RNSP as the reference gene as per suggested from the protocol, and the CT values obtained were exported to copy caller software to analyze the copy number calculated from each of the samples. For the mRNA expressions, we converted the RNA to the cDNA through reverse transcription and proceed to the FGF10 mRNA expression. The same method, we use the real-time PCR. So in this assay, we use the GAPTH as the reference gene. As we can see from the figure shows the value of the copy number calculated of FGF10 between normal and patients with NSCLP. We can see the green bar. The green bars represent the normal individuals, whereas the yellow bars represent the patients with NSCLP. So it was observed that the copy number calculated in patients with NSCLP was slightly higher, but not so significant compared to the control group, where the copy number calculated from of the patients is 2.29, whereas the normal is only 2.123, and the significant is 0.447. And then you can see from the results of the mRNA expression, where the figure shows the meanfold change of FGF10 between the normal and the patients with GAPTH as the reference gene. So it was observed that the expression of FGF10 was slightly higher in NSCLP patients compared to the normal individual, but also not significant, where we can see we observe that the fold change in the patients with NSCLP is 1.014. Whereas the normal only 1.0097. So the significant is 0.133. So not so significant in this result. And next, we do the correlation analysis, where we found that the R value is equal to negative 0.157, which is it suggested a weak negative correlation between FGF10 DNA copy number and mRNA expression in patient with non-syndromic cleft lip and or palate. So the conclusion. So this study has found that actually there was no significant difference between FGF10 DNA copy number and the mRNA expression with NSCLP among Malays. Besides, it was also revealed that there was a weak negative correlation between both variables with NSCLP. However, further investigation with a larger sample size and comprehensive analysis is warranted to determine the potential role of this gene in non-syndromic cleft lip and or palate among Malay patients. And then this is references that I use for this project, this acknowledgements. And I think that's all from me. Thank you.
Thank you, dear Noor, for your presentation. Dear guests, dear participants, have you any question for Noor?
So thank you very much for invitation, and not only to Sejad but all his entire team. I'm thankful. So my topic is, this is like a review, not research topic. So how to produce a recombinant protein using E. coli host. And let me know whenever my voice is not clear. So let me know. So why do we need protein? So you all know that proteins are used as a therapeutics, used as an enzyme for various industrial purpose, And for research also, we need good amount of protein with high purity to characterize that protein. Because you know that genome sequencing is easy, but to characterize all the proteins coded by that genome is difficult. So for characterization of protein also, we need good quality of protein, purified protein. So there are two ways to procure the protein, either purification from endogenous sources, or we can apply the recombinant DNA technology to produce recombinant protein. So when we purify the protein from endogenous sources, so there are serious limitations like quantity, purity, consistency. These are some of the serious problems. So scientists prefer to produce recombinant protein, means applying the recombinant DNA technology to produce good quality of protein, good amount of the protein. There are hundreds of recombinant proteins available in the market as a therapeutic, as a other process. I will not show you how to produce recombinant protein. I'm just showing why scientists prefer E. coli as a host for production of recombinant protein. So it's easy to culture, their life cycle is very short, well-known genetics, and cloning is technically easier. That's why most of the time scientists use the E. coli as a factory for production of recombinant protein because of all these good regions. But it has some problems also. Difficult or little expression of foreign genes. So it's not always true that when we get successful cloning, then we will get protein also in the E. coli. So sometimes we may not get expression, and we get sometimes very less expression. Post-translational modification also does not occur. Generally, glycosylations and other post-translational modification. If our protein requires glycosylation and other post-translational modification, then we cannot use E. coli as a host for production of recombinant protein. And no disulfide bond formation also. This is also a post-translational modification. So cytoplasm will not allow disulfide bond formation because E. coli cytoplasm is reducing. Its environment is reducing. And major problem in E. coli is inclusion body formation. Means our protein may get misfolded and forms a particle-like insoluble protein. So these are some of the problems. So we will discuss a different strategy. By applying that strategy, researchers can get good quality of protein, good amount of protein. So we have divided that strategy into 5 subtypes. So by changing the host strain, means nowadays there are several E. coli strains available. Depending upon your requirement, you can use that strain. So I'm going to discuss few strains. By changing the vector, as you know, there are different vectors having different affinity tag, and affinity tag also influence the solubility of your recombinant protein. Third, changing the culture parameter. By changing the culture parameter means we can add some ethanol or changing the temperature, culture temperature, and by co-expression, so we can co-express its interacting partner. I will discuss that also. And by altering the, so under 5 subheading, we will discuss the strategy to produce recombinant protein, means soluble recombinant protein in E. coli host. So most of the time, scientists prefer PET series of vector, PET 28A, PET 30, PET. So, and basic E. coli strain used for production of recombinant protein is BL21 strain, because BL21 strain is genetically engineered strain having lone and OMP protease deficient strain. Purpose is to stop or prevent the degradation of protein by protease. So scientists delete that gene, OMP and lone protease gene is deleted. That strain is called BL21. But when you use the PET28A vector having T7 promoter, that BL21 strain is not very suitable, and because T7 promoter of this vector will not be recognized by any of the RNA polymerase present in the E. coli strain. So solution of this problem is scientists have added D3 gene which codes for T7 RNA polymerase in that BL21. So that strain is called BL21D3. Means this strain is not only protease deficient but also it has T7 RNA polymerase gene, so that this RNA polymerase will bind to the T7 promoter present on the vector, and then gene transcription will start. But this strain have also one limitation, that is leaky expression. Leaky expression means when there is expression without inducer. So leaky expression sometimes not good. If your protein is toxic to the E. coli, so in case of leaky expression, that E. coli will not grow, means their number will not increase due to toxic effect. So to solve this problem, again, scientists have designed second strain, and that strain have, that is called BL21 pre-lyse strain. Means in the BL21D3, they have added the plasmid having lysozyme coding gene. So lysozyme is inhibitory to the T7 RNA polymerase, means the region of this you can say leaky expression is there is basal level of presence of T7 RNA polymerase. So to mask this, to inhibit this basal level of T7 RNA polymerase, scientists have added the plasmid having a lysozyme coding gene, and lysozyme is inhibitory to T7 RNA polymerase. So residual level or basal level of T7 RNA polymerase will not bind, or you can say it is inhibited by T7 lysozyme. So it is not available to bind here. So there is no leaky expression. So if your protein is toxic to the E. coli, so you should not use BL21D3, but you should use BL21D3 pre-lyse strain. So this is second strain we have discussed. It has very less leaky expression or no leaky expression. Now, most of the time when we use the eukaryotic gene for expression into the E. coli to produce the recombinant protein, so there is codon biasness. So codon biasness is, you can see that there are synonymous codons for a particular amino acid. So not all organisms doesn't use all the synonymous codon equally. Some organism prefer some codons, some different organism. So you can see from this data that AGA codon means arginine is coded by 6 synonymous codon. So AGA is present in 22% of cases, while in case of Drosophila 10% and E. coli 1%. Means E. coli doesn't prefer AGA. E. coli doesn't prefer CGA, but human they prefer. So what will happen? This different preference of synonymous codon is called codon biasness. So how it will affect production of recombinant protein in E. coli? So if we are using the gene from different organism, so due to codon biasness, either translation will stop or rate of translation will less. So ultimate result is either you will get truncated recombinant protein or you will get very less amount of protein. Despite of everything is good, you may not get a sufficient amount of recombinant protein due to codon biasness. So the region of codon, ultimate codon biasness is presence of rare tRNA, less amount of tRNA, which bind to the rare codons. Means the codon which is not preferred by E. coli is called rare codon. So concentration of tRNA is also very less. So during translation, availability of that tRNA is less. That's why translation rate will go slow down, or sometimes translation will stop. So to solve this problem, this strategy company have created again the strain. So they have added extra copy of rare tRNA gene in the E. coli, and that strain is called codon plus strain. So there are 5-6 rare codons which are not preferred or very less preferred. So they have prepared 2 strains. One is called BL21 codon plus RIL, and second is BL21 codon plus RP strain. Means they have created 2 strains. So in both strains they have added different sets of rare tRNA. So you have to choose whether your gene is AT rich or GC rich. Accordingly, you can use this strain, either BL21 codon plus RIL or RP. So second company, that Novagene, they have created Rosetta strain. Means they have added all the 6 rare tRNA gene in the BL21 strain, and that strain is called Rosetta strain. Means if you are facing a problem of codon biasness, means you are not getting the sufficient amount of protein production in E. coli due to codon biasness, you should use Rosetta strain, not a normal BL21 strain. Otherwise, you will not get protein production or less protein production. Transcription will be fine, but protein translation will be hampered, translation will be less, so protein production will be less. So you should use Rosetta strain. There are so many mutant strain also produced, self-mutant strain, that is BL21, C43, C41. So you can produce toxic protein also in C43 strain, C41 strain. Means generally this transmembrane proteins, globular proteins are toxic. But this is self-mutant strain produced by, you can say, this group. So, and this strain is supplied by leucine. So you can produce initially production of membrane protein and globular protein was difficult, but you can use this strain to produce globular protein, which is toxic to the E. coli. But this is mutant strain which will allow, which will tolerate the toxic effect of the protein also. I'm not going to discuss the mechanism. So based on that self-mutant strain, knowledge of the self-mutant strain, this NEB company, New England Biolab, have created this LEMO21D3 strain, in which you can see T7 RNA polymerase cloned under titratable ramenose promoter, so that you can control the level of expression, and that is the basis for tolerating the toxic effect of the protein. So if your protein is toxic, globular protein or membrane protein, you can use this LEMO21 strain. Arctic Express strain. So this strain have, you can see here, cold adapted chaperone gene has been added, because scientists have observed that when they reduce the culture temperature after induction, so they are not able to see the soluble protein, because when they reduce the culture temperature, normal chaperone activity also reduced. So that chaperone is not able to properly fold the protein. So scientists have added cold adapted chaperone coding gene in this strain, and that is called Arctic Express strain. BL21 star. So RNA EU131 gene mutation confers high stability of mRNA. So as you know, mRNA may also get degraded. So if you are facing this problem, you should use BL21 star strain, in which this RNase coding gene has been mutated. Expression of protein which requires disulfide bond. As you know, most of the proteins require disulfide bond for its proper folding. If disulfide bond will not form, then it will not retain its natural native conformation. So you will not get enzymatic activity or protein activity. So E. coli cytoplasm will not allow, normal E. coli strain, their cytoplasm will not allow production of disulfide bond because of reducing environment, and disulfide bond formation is oxidation. So there are two solutions. Either you transport your protein into the periplasm of E. coli, or you should change the cytoplasmic environment from reducing to oxidizing. So scientists find that this thioredoxine reductase and glutathione reductase enzyme are responsible for creation of reducing environment of the E. coli cytoplasm. So they mutated this two gene so that cytoplasm, E. coli cytoplasm will not remain reducing. So if you wish to produce the protein which requires disulfide bond formation, you can use origami strain. This mutant strain having this mutation, thioredoxine reductase mutation and glutathione reductase mutation, that strain is called origami strain. So again, scientists observed that if there are multiple disulfide bond formation required in the protein, so this only environmental condition is not sufficient, because it can form a random disulfide bond formation. So origami strain is not good, because it will only allow disulfide bond formation, but it will not control whether this disulfide bond is proper or not. So they have added in origami strain, they have added this disulfide bond isomerase gene, which will only allow the proper disulfide bond formation, proper combinations of disulfide bond formation. So you should use shuffle strain in which that disulfide bond isomerase has been added. If your protein requires more than one pair of disulfide bond, then you should use. Otherwise, origami strain, if there is only 2 cysteine in your protein, so it will only form one disulfide bond, no need of shuffle strain. So we have discussed rapidly about different E. coli strains available in the market to produce recombinant protein, depending upon your requirement, whether you are facing codon biasness or your protein is toxic or your protein. So, but still, scientists are working on production of glycosylated protein also in the E. coli, but that strains are not commercially available as per my information. But scientists are trying to produce the genetically engineered E. coli strain, which will allow glycosylated protein also. So this is summary of all these strains. You can find this in my now. Second strategy is changing the vector, as you know. So I'm going. So we have discussed PET28 vector. You know that PET28 vector have very small tag, affinity tag, histidine tag, which is preferred, which is very low. Yes.
Excuse me, sir.
Yes, sir.
Sir, can you kindly share your slides again? They are not visible.
It's not showing.
Sir,
Slide is not
share.
visible. Slide is not visible.
Yes,
Hello?
sir.
Oh, okay. Why it is not showing? Just again, wait. Now?
Yes, sir. Thank you so much, sir.
Okay. I don't know what happens. Huh? So up to this level, it was showing this. Hello. This is summary of all the different strains available in the market, so you can utilize this strain for production of recombinant protein depending upon the problem you are facing. Now, second strategy is by changing the vector. As you know, PET vector is preferred, PET28A, because it has histidine tag, which is very small affinity tag and very robust affinity tag, but it has some limitations also. So histidine tag reduce the solubility of your recombinant protein. So it has been observed that if your recombinant protein has histidine tag, so sometimes it will form an inclusion body. So inclusion body is not preferred because it is insoluble protein. You cannot use this protein for enzymatic activity or other activity. So if you are facing the problem of inclusion body due to histidine tag, you should change your vector. Means you should clone your vector in the you should insert your gene of interest into the vector having affinity tag, which will not hamper the solubility. So you can clone your gene in the PMAL vector having MBP tag. MBP tagged protein or MBP increase the solubility of the conjugated protein. But it has also limitations. You can see its size is very large, 44 kilodalton. So sometimes if your protein is very less, then you cannot allow that long tag to conjugate with your protein. That's why we don't prefer. But if there is problem of solubility, you can use this. There are again different vectors. You can use this news PET20, PET44, PET43 having an utilization substance tag, which also increase the solubility of your protein. You can also use p-hat vector. So here in p-hat vector, histidine amino acid is not continuous. So due to this discontinuous presence of histidine, it will not decrease the solubility of your protein. Means tag affinity tag is as it is, but due to non-continuous presence of histidine, it will not decrease the solubility of your protein. So this is, you can see here. So here histidine is not continuous, while in case of PET vector, histidine was continuous, 6 histidine, but here there is no so due to this non-continuous presence, it will not reduce the solubility of the protein. PGX vector. So if you need to change the affinity tag, you can use this PGX vector. So your protein will have glutathione S transferase affinity tag. It also increases the solubility of your protein, and size you can see 25-26 kilodalton. So depending upon the problems of solubility, you can change the tag, and accordingly you can use the vector. TQ vector. So gene is cloned under T5 promoter. As you know, in PET28 vector, you have seen T7 promoter, and that cannot be, that T7 promoter is not recognized by any of the RNA polymerase present in the E. coli. But here T5 promoter is also viral promoter, a strong promoter. It can be recognized by E. coli strain. So no need of BL21D3 strain. Here a strain expression strain is used, M15 and SG1003, and this strain possesses PREF4 plasmid. This plasmid encodes lac repressor, which ensures tight regulation. So there is no leaky expression also. If we are using this PQ vector system, there is no chances of leaky expression. SUMO, so solubility of conjugated protein increases if we are using this vector. And one advantage of this vector is we can precisely delete the affinity tag. So here, if you are not sure about your vector, and if you wish to again think you have to subclone your gene of interest, so initially you can check this MCS of different vectors so that you can subclone your gene of interest into different vector just by cutting the that gene, inserted gene, and inserting. That is called subcloning. But that restriction site which you are going to use must be similar in different vector. Then only you can easily subclone. Otherwise, what will happen? Again, you have to design a primer according to MCS, and then again you have to do PCR, everything. If restriction site is common in second vector, then directly you can cut your gene of interest and insert into next vector. So you can do this exercise. So these are some of the affinity tag and their length. You can change the physical or chemical agents also if you are facing the problem of solubility. You can culture a recombinant host at low temperature. So purpose of growing the E. coli at low temperature after induction is, we think that if we grow that E. coli at low temperature, so rate of expression will be less. So rate of expression is less, so folding of that protein will be taken care of by the chaperone protein present in the E. coli. Otherwise, what happens? If rate of production is high, so chaperone protein present in the E. coli will not take care of folding of your protein, so your protein becomes inclusion body. Second is ethanol addition. So it has been also observed that 3% ethanol addition increase the solubility of your protein. Heat shock, salt addition. So different reagents, scientists, different group of scientists have observed that it has some effect in increasing the solubility of protein as well as increasing the production of protein also. So you can try. Suppose if you are not able to purchase that strain, E. coli strain, if you are not able to purchase different host, different vector, you can try these chemical agents also. After induction, you can add. So I have written that they cited that reference also. Fourth, co-expression. So what happens? Desired gene co-expressed with different combinations of chaperone showed promising result. Means one of the reason of formation of inclusion body is not proper folding of your protein. So as you know, chaperone protein is responsible for folding of the protein. But if your production rate is high, so chaperone protein present in E. coli will not able to fold your protein properly. So one of the thinking is, if we co-express chaperone gene also, then that chaperone will take care of our protein folding. And then scientists have observed a good result also by adding co-expressing the chaperone gene along with your target gene. Second, co-expression of interacting partner of the complex. Suppose your target protein is X. You are trying to produce that X protein alone in the E. coli. But in native condition, that X protein remain associated with Y protein. So due to that association, their hydrophobic patches are closed together. So hydrophilic patches are remaining in aqueous environment. So that protein will remain in soluble form. But when you are producing alone, so their hydrophobic patches are exposed in the aqueous environment. So there is chances of precipitation or inclusion body formation. So one of the you can say way to get the soluble protein is you try to co-express their interacting partner protein. So you co-express both genes. If you know the character of your protein, which are associated with other protein in its native condition, native host, you can co-clone both genes and co-transform and co-express in individual E. coli strain. Sometimes people get good result when they co-express. Fifth, changing the gene sequence. So sometimes removal of signal peptide coding sequence from ORF, it also increase the production rate, because you know that purpose of signal peptide is transportation. But if your target is only to check the characterize or use that protein for its enzymatic activity, so no need of signal peptide in E. coli. Here you don't wish to transport your protein in the mitochondria or other compartment. So why not you only clone the gene corresponding to the mature protein? Why signal peptide you will include? Because signal peptide may hamper the production of the protein. Because it is extra. Removal of highly hydrophobic stretches from the protein. So sometimes, how much time we have? Okay, hello. I have rapidly gone through. I will take questions also. Hello, are you listening? Am I audible? Hello.
Yes, sir.
Okay, okay. How much time left?
Okay, if you want, you can finish.
Okay, 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 okay. So we can remove the hydrophobic stretches from the protein. Means if your target is only the exposed part of the protein from the membrane, not the embedded part, because embedded transmembrane embedded part have hydrophobic nature, and you are going to produce that protein in E. coli cytoplasm. So due to hydrophobic state, your protein may form inclusion body. So why not you should only clone the gene corresponding to the hydrophilic patches. So there is chances of producing the soluble recombinant protein. You can optimize that gene sequence also, because you know that there are so many post-transcriptional regulation occurs. But here you wish to produce more amount of protein. So if you think that that RNA level regulation, if that RNA forming a secondary structure which will not allow direct participation of that mRNA into translation. So why not you remove that region so that your RNA will not form that structure, which will hamper the initiation of the translation. Because you know that if RBS, signed Dalgarno sequence, is not free, then RNA this ribosomal smaller unit will not bind, so translation will not start. So despite the presence of RNA, mRNA, you may not get protein in that E. coli. So you should remove that part. You can optimize that sequence, and you can also change the rare codon, which is hampering the production of your protein. So you can change rare codon to the preferred codon, favorable codon, by site-directed mutagenesis. Means you can change the gene sequence also if you know the region of not producing the sufficient amount of protein or soluble protein. So this is overall about different strategies which can be utilized for production of good quality of protein and soluble protein. Amount is also important, and their solubility, three-dimensional structure, proper folding is also required. So this is overall strategy. If you have any question, please. I hope I have completed within 25 minutes. Right? Hello. So you can ask question. Thank you again to all the organizers, all the team. please ask question if you have any question, any suggestion. Yes.
Dear doctors, thank you very much for your presentation. Dear guests, have you any question?
Yes. No question?
Not, yes,
Dr.
maybe.
Dr. Gopal, you were talking about E. coli.
Yes.
You were talking about some mature proteins. Can you specify some mature proteins?
Mature protein means, as you know, signal peptide is utilized for transportation of protein. And
Okay.
so even in case of, so I have cloned Helicobacter pylori type 4 secretion protein. So signal peptide is utilized for transportation. So we have only cloned the remaining part by removing the signal peptide coding. Suppose 21 amino acid is a signal peptide. So we cloned the gene corresponding to starting from 22nd amino acid. Are you getting?
Yes, yes. Thank you very
Yes.
much. Thank you very
Yes.
much.
Yes.
Uzman Ahmed, had you any question? So because I see your raised hand.
Yes. So you can connect with me through social site. I'm using LinkedIn and Facebook, ResearchGate. So you can connect even in future if you are facing problem in production of recombinant protein using E. coli host. You are welcome to ask question. You can send your WhatsApp message also. So
thank you so much, Dr. Gopal, for your presentation. It was really excellent one. I just wanted to know that how MBP proteins or maltose binding proteins or that these tags can increase the solubility of the recombinant proteins.
see, you know that some proteins, since this is 44 kilodalton protein, है ना? So
yeah, it is quite big protein, and
yes, so it is more soluble. So as you know, if two persons are, one is good, one is bad. So good person may influence the bad person sometimes. So continuous histidine is decreasing solubility, and this MBP and GST tag, they increase the solubility because it is conjugated. So it increase the solubility. Exact mechanism, I don't know exact mechanism how it affects.
Oh, okay, thank you. You mean that it's the influence rather than something else? Okay, thank you so much. Thank you.
Okay, thank you, Doctor, very much for your again presentation, and thank you for your question, dear presenters. Okay, next, Dr. Majida, are you ready?
Myself, Dr. Majida Rashid from Department of Life Sciences, Khaja Fleet Engineering University, Rahim Yar Khan, Punjab, Pakistan. This is a collaborative study and a campaign, an awareness campaign, basically, and solution techniques dissemination for the proper disposal of dead animals to clean environment from hazardous microbes. A case control study in provinces of Pakistan in the year 2023 to 2024. Focus of this research is to highlight an issue expected to create by not disposing, burying dead animals in proper focus of this research is to highlight an issue expected to create by non-disposing, burying dead animals in proper way. It may create high level increase in environment, deadly microorganisms, and these non-disposed animals may also affect birds population like vultures, which are now at threatened level of extinction. Without vultures to consume the carcasses, the dead bodies of animals remain untouched, accumulating decaying remains, and it is crucial for the ecosystem and general public health. So there is dire need to increase information and to implement suitable disposal techniques to reduce the risk of hazardous microorganisms and to assure safety of birds. The study is being conducted in whole Pakistan from 2023 to 2024. Using a case control study methodology, regions with bad disposal habits were chosen as cases, where regions with good habits were chosen as control. Total of 100 awareness campaigns, 500 interviews, and 100 different site visits and conference presentations were done to deliver and gather information to evaluate present dead animal disposal techniques, and vultures population decline is also being recorded. The result of this study highlight the importance of an effective awareness campaigns in attaining the objectives. The study's conclusion underlined the presence of dead animals in the region and ubiquity of unsafe methods for disposing of dead animals and the dangers of microbial contamination that go along with them. Compared to locations with suitable disposal, those with improper disposal had a higher presence of dangerous microorganisms in the environment and loss of vulture population. To support these findings, statistical data was examined. This case control study concludes by emphasizing significance of resolving the issue of improper animal disposal in all regions of Pakistan. Burial and disposal of dead animals and what factors involved in the death of animals. This is very important to understand. Factors are natural death, diseases, accidents like vehicle collisions, human caused deaths, hunting, being shot because of multiple reasons, predation, animal caused death, deaths during fight with other animals, starvation, and what are environmental hazards created by these dead bodies? Dead bodies of animals can cause pollution in the environment. Dead bodies of animals can cause several diseases that are detrimental or even deadly for human or domestic animals and bird population of that area. And leptospirosis is a disease that transfers to human through the handling of an infected dead animal. Decaying matter of dead animals may mix with water, contaminate it, causing serious health issues for those who drink it. Bacteria that grow on these dead bodies may enter in the air and cause diseases in humans of that area. On the other hand, these bare dead bodies may spread insensitivity in the people. So there are some methods to dispose of dead animals. All dead animals should be handled only while wearing gloves. A carcass should be placed in a plastic body bag and sealed as soon as possible. And one should avoid direct contact with the dead animal's body fluids, avoid contact with the dead animal's external parasites. Proper disposal of carcass is critical to prevent exposure of other animals and humans to disease. Three common effective methods of carcass disposal are incineration, burying, rendering. Incineration is the preferred method to use when carcass is deceased, and acceptable alternative is to bury the carcass. So persons who have direct contact with animals, especially carnivorous animals, on the regular basis are highly recommended to receive the rabies pre-exposure vaccination series. And this campaign is very much important to deliver and to understand, as there are a lot of issues world is bearing, like climate change, biodiversity, and ecosystem disturbance, and such like things may be the reason for that. Thank you very much.
Thank you very much, Dr. Majida, for your presentation, for your talking. Have you any questions, dear guests? Okay, if you haven't any question, I will try to invite as a speaker. Again, thank you very much, dear Dr. Majida.
Thank you, ma'am.
Dr. Kavita, are you ready? Dr. Kavita, do you hear me? Okay, if you're ready, you can start, please.
Yes, good evening, ladies and gentlemen. My name is Dr. Kavita. I am delighted to be here with you today at the online international conference on biological and medical sciences 2023. As an assistant professor of mathematics at Sri Chandrasekharendra Saraswati Vishwamahavidyalaya deemed university in Kanchipuram, it is an honor to have the opportunity to share my knowledge and insights with this esteemed gathering of researchers, scholars, and expertises in the field of biological and medical sciences. Throughout my academic journey, I have been passionate about exploring the intersection between mathematics and the life sciences. My research interests lie in developing statistical models and mathematical approaches to analyze complex biological data, particularly in the field of genomics. I firmly believe that the application of mathematics and statistical methodologies can unravel the hidden patterns within genomics data and unlock vital information about the secrets of genome. As an enthusiastic advocate for interdisciplinary collaboration, I have had the privilege of working alongside brilliant researchers from diverse backgrounds, including biologists, bioinformaticians, and clinicians. Together, we have embarked on fascinating projects aimed at understanding the intricacies of biological processes, identifying genetic links to diseases, and ultimately contributing the advancements in healthcare and personalized medicines. Today, I stand before you with a profound sense of excitement and anticipation. In this session, I will be presenting on developing statistical models for analyzing genomic data, unraveling the secrets of genome. We will explore the power of statistical methods in making sense of the vast amount of genomic data generated through high throughput sequencing technologies. By the end of this talk, I hope to convey the significance of statistical genomics in shedding light on the complexities of the genome and its pivotal role in shaping life as we know it. Once again, I express my gratitude to the organizers of ICBMS 2023 for providing me with this platform to share my research and ideas. I am eager to engage in meaningful discussions, exchange knowledge, and learn from the expertise gathered here. Thank you for your attention, and let us embark on the existing journey of unraveling the secrets of genome together. Imagine a world where we can decode the secrets of life hidden within our DNA, a world where sophisticated statistical model unlock the mysteries of the human genome, leading us to personalized treatments for diseases, groundbreaking insights into evolution.
Dr. Kavita, sorry, can we see other slides? Okay.
Yes, sir.
Now,
Okay.
Now is changeable.
Okay.
Thank you.
Yes, imagine a world where we can decode the secrets of life hidden within our DNA, and a world where sophisticated statistical models unlock the mysteries of the human genome, leading us to personalized treatments for diseases, groundbreaking insights into evaluation, and a deeper understanding of our very existence. Today, I stand before you to talk about how we are turning this vision into a reality through the development of statistical models for analyzing genomic data. Imagine a world where we can unlock the secrets of our genes swiftly and accurately. Have you ever wondered how the rapid advancements in sequencing technologies are reshaping genomic research? Genomics is the study of an organism, a complete set of DNA which holds the blueprint of life itself. Recent advance in high throughput sequencing have revolutionized our ability to decode this complex genetic information at an unprecedented scale. Here, the aim of this research, the research project seeks, actually these are the datas. Thanks to these advancements, we are now able to generate vast amounts of biological data with incredible speed and accuracy, opening doors to previously uncharted territories in genetic research. The significance and relevance. Do you know why understanding genomic is essential and how it impacts various aspects of life sciences? Unrevealed. The intricacies of the genome is not merely an academic pursuit. It holds the key to unlock critical insight into disease development, personalized medicine, and even the potential to address previously untreatable conditions. Access to cured genomic data is crucial for researchers and educators alike. These are the websites. We can get a free data available in those websites. Here one more thing, more data are available in here, but remember to review their data usage policies, licensing, and any specific requirements for accessing to using these data. Okay, so if you are looking for the genomic data for research or educational purpose, I recommend checking publicly available database and repositories that host curated genomic data. So some well-known public genomic databases only given here. If you want, you can take the screenshot of this. It's a very useful website. So these well-known public genomic database provide a vast array of genetic information that can unlock new insight into biological and disease. So make sure to review their data usage policies, license, and any specific requirements to access. Okay, utilize it and do your research progress. Okay. The next one. I want to provide a brief teaser of what you can expect from my presentation. Throughout this talk, we will explore the cutting edge statistical approach that have enabled us to make unprecedented strides in genomic research. Get ready to dive into a world where data and statistics converge to unlock the secrets of life. Do we ready? Yeah, yes. Now I want to show you that omic logic bioinformatics education. Can you see the screen? Can you possible to see the screen? Omic-Logics. It's a maybe you biological people can know this. First of all, you need to register. It's a free website only. It's a very useful and very well known, very interesting website for the researchers. So please, first of all, you have to log in this website. I already logged in. So register for free, you have to use here. And many courses are there. Actually, I belongs to mathematics department. How can I know the protein genomes and all means? From this website only. It's very useful and interesting and a very easily understandable website. Here we have more courses about the genomic data we need means we just type genomic. I think it's very, very useful to the researchers. All type of biological data are here. Bioinformatics data are here. More of the biological people are here. You may feel better for this from this website. These are the genomic course. It's actually a genomic course. You can learn everything from here. Here, introduction to genomics, DNA structure variants. We can learn everything here with the visual data. Ladies and gentlemen, as we dive deeper into the fascinating world of cancer, genomics, and the identification of a driver mutation, I would like to introduce you to the valuable resource that can gently aid your research endeavor in this field. Allow me to introduce this Omic-Logic website. Here, this is the Omic-Logic website. Okay, Omic-Logic is an innovative and comprehensive platform that empowers researchers and clinicians with cutting-edge tools for analyzing and interpreting complex genomic data. It is designed to streamline your journey through the vast landscape of omics data analysis from genomic to transcriptomic and beyond. Okay, this user-friendly platform offers a wide array of features including data integration and analysis, statistical model and machine learning, pathway and network analysis, interactive visualizations, educational resources. Here, look at this mutability analysis interpretation, differential mutation analysis. You just go to the learn means it can show some of the lessons and it can deliver the lecture to you. More genomic patterns are here, more genomic courses are here. So please use this website and you complete your research well and good. Okay. And one more thing, data analysis using R, big data analysis using R or Python, everything you can learn here. This is bioinfo in Python. So you can go here means you can get more codings, Python codings and R codings you need. Now you just go to R coding courses. Getting started with the bioinformatics means you can get more codings to analyze the big data and you can get more 3D effects, 3D effects and all you can do it using this R coding. So every R coding are here. R coding courses gives you the detailed study about the R. Okay, I think you can clear with this. Pathway and network analysis are there. Interactive visualizations are here. Visualizing complex genomic data is more simple with Omic-Logics. interactive and initiative visualizations facilitating a deeper understanding of your findings. So educational resources are there. So as we continue our exploring of identifying driver mutation in cancer, I encourage you to explore the Omic Logic website to enhance your research capabilities and gain valuable insights into the world of omic data analysis. Okay, you can access the Omic Logic website through the provided URL or scan the QR code on the screen. Take advantage of this powerful resource to advance your cancer genomic research and contribute to the understanding and the treatment of the cancer. Okay. Thank you for your attention, and I welcome any question or discussion about Omic Logic and its potential applications in your research endeavor. Let's embark on this exciting journey together, utilizing advanced tools to unlock the secrets of cancer genomics. Okay. This research project seeks to make significant contribution in the field of genomics. The main objective of our research connecting it to the broader context of genomic and high throughput sequencing. So today I am excited to share with you the pivotal role that sophisticated statistical models play in harnessing the power of genomics data and how they help us discover hidden patterns that hold the potential to revolutionize medicine and biological research. The genome is the complete set of genetic material or DNA present in an organism. It serves as a blueprint of life, containing all the information necessary for an organism's growth, development, functioning, and reproduction. Is it? The genome is organized into structures called chromosomes, and each chromosome contains thousands of genes, which are the functional units of heredity. Okay. And so the chromosomes is the functional units of heredity. What is the role of the genome in biological process actually? The genetic heritage, the role of genomic in biological process is genetic inherent, protein synthesis, cell regular and differentiation, cell replication and repairs, response to environmental stimuli, evolution and adaptation. These are the role of genome in disease biological process. In disease development also the role of genome is there. Whatever the disease development, genetic diseases are there. Okay, some disease are directly caused by mutation or alteration in specific genes. Is it? These genetic diseases are inherited from parents or occurred spontaneously due to mutation during development. So, genetic diseases are some diseases, and susceptibility to diseases. Certain genetic variants can increase an individual's susceptibility to certain diseases. This variation may interact with the environmental factor or influence disease risk, like the normal cold. Okay, so this is happen with environmental factors. Cancer, the genetic mutation can lead to uncontrolled cell growth under divisions, resulting in cancer. Is it? So the specific genes known as oncogenes and tumor suppressors gene are involved in the regulation of cell growth and are often implicated in cancer development. Is it? So next, infectious diseases. whatever the infectious diseases, genetic factors can influence an individual's susceptibility and response to infectious agents, affecting the course and severity of the infectious diseases. So understanding the genome role in biological process and disease development is critical for advancing medical research, developing target therapies, and improving overall health care. High throughput sequencing technologies have played a pivotal role in advancing our knowledge of the genome and its implications in various aspects of biology and medicine. Is it? So now some common statistical analysis used in genomes. Whatever the common statistical analysis we can use, descriptive statistics like mean, median, mode, that and all the mean, median, mode, standard deviation, and the percentiles, that and all the basic descriptive statistics. And the differential expression analysis means the gene expression levels between differential conditions or groups to identify genes that are differentially expressed or not, that we can find using this differential expression analysis. Okay. And then the third one, gene set enrichment analysis. In the gene set enrichment analysis, we can determine whether the predefined sets of genes are enriched in differentially expressed genes compared to what would be expected by chance. Okay, so the next statistical analysis is principal component analysis, PCA. The principal component analysis is used to reduce the dimensionality of gene expression data and visualize the data in the lower dimensional space, making it easier to observe the patterns and clusters. Okay. So it used to reduce the dimensionality. Okay. Next is cluster analysis. In this cluster analysis, the groups of genes are samples with a similar expression profiles into clusters, helping to identify co-regular genes or distinct subgroups within the data that we can analyze. Okay, using the cluster analysis. Next, association studies. In genetics, association studies test for correlation between genomic variation and specific traits or diseases to identify genetic makers associated with a particular phenotypes that we can analyze using the association studies. Okay, so the genome, next one, the genome-wide association studies. In this genome-wide association studies assesses the relationship between millions of genetic variants across the genome and tries to identify genetic association with diseases or traits that we can analyze. Okay, with diseases or traits that we can analyze. Next, network analysis. In this network analysis, this helps to study the interactions between genes and proteins. Okay, so network analysis used to study the interaction between genes and proteins. Uncovering functional relationship and pathways are analyzed using this. Okay. Next, time series analysis. For what purpose we are using time series analysis? Time course data. Time series analysis is used to identify genes with a dynamic expression pattern over time. So, expression pattern of gene over time we can analyze here. Next, survival analysis used to this survival analysis applied with when studying time to event data. Okay, such as the time until the specific outcome occurs. Okay, time until the specific outcome. Until the specific outcome. And for example, time to recurrence in cancer patient. Okay, that in that case, survival analysis very useful. And multiple testing correction, The genomic data often involves testing numerous hypotheses simultaneously, requiring adjustments for multiple testing to control the false discovery rate. So it's important to use appropriate statistical methods and tools for the specific genomic data and research questions at hand. Okay, so research question, first you have to analyze and use the particular or appropriate statistical method. Okay, many of these analyses are performed using specialized bioinformatics software and programming languages like R or Python. This R or Python you can learn using those that website I shown you. Okay. Along the various more codings are also available there. So please use that website. Okay. It's a free only. It's a user friendly and a free website only. Okay. Along with various bioinformatics libraries and packages. Additionally, it's essential to consider data pre-processing and normalization steps before conducting statistical analysis to ensure reliable results are there. Okay. First, you can ensure. Yes.
So, Dr. Dr.
Yes.
Kavya, the time is and see any slide now.
Oh, okay.
And also time is over.
Oh, okay, ma'am. Okay, ma'am. On our 2 slides. Wait a minute. The statistical model applied to genomic data can be an informative and engaging expertise. Here is the general outline and key points you can include for each of the topic. Okay. And these statistical models are used to identify biomarkers for cancer diagnosis, prognosis, and treatment response. Okay. And next, identify the driver mutation in cancer. In cancer genomic research, one important this is where our sophisticated statistical model come into play. They provide us with a systematic and objective approach to analyze genomic data. By applying this model, we can effectively identify patterns and trends in the vast sea of genomic information, helping us pinpoint those elusive driver mutation. Okay. The impact of identifying driver mutations goes beyond just understanding cancer biology. It also opens up new possibilities for targeting therapies. By targeting the specific genetic alterations responsible for driving cancer growth, we can develop more effective and personalized treatment for patients. Okay. So these are the statistical models come into play in this application. Mutation analysis, mutation frequency, mutation hotspot. Mutation hotspot, functional impact, pathway, and network analysis integration with clinical data. Okay. So now I come to my conclusion. The field of genomics coupled with advanced statistical modeling has opened up new horizons for research and application across various domains. Okay. So the field of genomes coupled with advanced statistical modeling. Okay. In cancer, genomic statistical model have been instrumental in identifying driver mutation and potential therapy target, paving the way for personalized cancer treatment. Additionally, the application of statistical techniques in pharma has revolutionized drug development, ensuring more effective and tailoring therapies based on the individual's genetic profile only. Okay, individual's genetic profile only you can. However, challenges remain, including handling the vast volume of genomic data, addressing issues of data quality and standardization, and developing models robust enough to handle the inherent biological variability. Interdisciplinary collaborations between mathematicians, statisticians, biologists, and clinicians will be crucial in addressing these challenges and advancing the field. Okay, because I am a mathematician. In conclusion, the synergy between genomic and statistical modeling holds immense promise for tackling fundamental biological questions, transforming medicine, and address global challenges. By continuing to innovate, the collaborate researchers can unlock the full potential of genomic data, sharing in a new era of precious medicine, sustainable agriculture, and a deeper understanding of life intricacies. So thank you so much. These are my references: bioinformatics and computational biology solution using R and a bioconductor is the main reference here. Using R programming, more codings and more informations are there in that book. And the statistical analysis next generation sequencing data is very useful to understanding the genomic data. Okay, so thank you for giving this opportunity. Thank you so much.
Yes, you can start.
Okay, Assalamu alaikum everyone. My name is Laiba Asim. I am from University of Central Punjab, Lahore, Pakistan, and I have completed my research under the supervision of Dr. Salma Batul from University of Central Punjab and under the co-supervision of Dr. Rahman Shahzeb Salim from Lahore University of Management Sciences. The title of my MS project is Design, Synthesis and Assessment of 3-picolinic acid-based siderophores in relation to mineral uptake and plant growth. So let's start with the background. Siderophores are basically iron chelators that are produced by bacteria, fungi, plants in order to sequester iron from the environment. And as iron is most abundant form present in Earth's crust, but it has low bioavailability. So siderophores facilitate iron uptake, and there are different types of strategies for iron uptake. One is the reduction based, and another one is a chelation based. In our study, we target rice, and as it utilizes strategy 2 for uptake of iron, in which plants produce siderophores and release them into the soil, and siderophores there form a complex with the iron and then transport it through different proteins and make it available to the cell. The objectives of the study include chemical synthesis of siderophores, the characterization, biological assessment of synthesized siderophores on rice, and at the end, computational analysis to validate results of biological assessment. Moving towards material and methods, for the synthesis of siderophores, 3-picolinic acid reactions with different amines were carried out in order to yield amide. Then for the characterization, FTIR was employed as it identifies chemical bonds in the molecule by producing an IR absorption spectrum. And then NMR was done as it determines the content and the purity of the sample as well as its molecular structure. After characterization, biological assessment was done on rice, in which we optimized the soil first. Then phenotypic screening was done, followed by the measurement of plant growth parameters, including height and diameter of shoot. And then for mineral uptake analysis, chlorophyll quantification was done first, and then ICBMS, which is inductive coupled plasma optical emission spectroscopy, was done to get quantified results of mineral uptake. At the end, computational analysis of phenotypically screened siderophores was done with the transporter proteins of siderophores in rice. For this purpose, molecular docking tool called Pyrex was used, and then BioVia Discovery Studio was used to get its simulation. Moving towards results, here are the results of the chemical synthesis. These are the structures of the compounds that we get when we react 3 picolinic acid with different amines in order to yield amide. All the reactions were based on EDC coupling and carried out at room temperature overnight, and the reaction progress was monitored via TLC. And TLC suggested us to do column chromatography in order to yield the purified product. The synthesized hydrophores then successfully characterized using FTIR. The FTIR here clearly shows the formation of amide as a long stretch was shown from 3055.4 to 1650.94, and all the essential peaks of the function groups are there that shows the formation of amide. Now, after confirmation of function groups of FTIR, we opt NMR to get verification about the purity and the molecular structure. The proton NMR of the respective compound that actually shows the number of hydrogens present in our structure. After successful completion of two objectives, we move towards a biological assessment, in which the first step was very crucial, that is soil optimization. But after 12 experiments, we get our desired optimized soil in which iron deficiency symptoms were cleared because of the increase in pH and due to the use of sodium hydroxide and calcium carbonate in combination. Here is a visual documentation of phenotypic screening of our synthesized hydrophores with a control, and as they clearly show better results as compared to control, which is just simple tap water. The plant growth analysis showed that 009 has greater height as compared to 006 and the control, and 009 have greater diameter that was measured using digital Werner caliper. Then the chlorophyll quantification was done that also shows that 009 have more chlorophyll content supported by increased growth and increased diameter. At the end, ICBMS was done that actually quantifies the mineral content. The result showed that 009 increase iron uptake as compared to other essential minerals, as well as significantly increased content of iron from control. At the end, result validation was get using molecular docking by Pyrex that reveals that 009 have more affinity towards citrophore transporter because lower value indicates the better results. And it also revealed that amino acid that are present at the ligand binding site. Coming towards the discussion, among all the synthesized hydrophores, 006 and 009 were phenotypically screened as they show green and healthy phenotype as compared to control. And when they were analyzed further, we came to know that 009 have increased high diameter of shoot supported by increased chlorophyll content, and ICPOES also revealed that all is due to increased uptake of iron. Moreover, Pyrex also revealed that 009 have increased binding affinity towards a transporter protein, and when the results were compared with the literature, we came to know that 009 has increased iron uptake as compared to PDMA, which is also a hydrophore that has recently been synthesized. And yes, our work do have practical importance as the study targets rice. So we can say that our end user is human, and it falls under SDG 2 and 13 that aims to end hunger and achieve food security by increasing nutrition and meet the increased food demand by increasing agriculture sustainability. Coming towards the main highlights of the work done, as it is the first study on design and synthesis of 3 picolinic acid-based hydrophores, and it targets complicated crop, that is rice, and all the results were displayed with the visual documentation. At the end, we can conclude that LARS 009 can replace chemical fertilizers and could become a reality in the near future. Thank you so much. This is all from my side.
My screen visible to you or not?
Yes. Yes, it is visible now.
Okay. Assalamu alaikum. I am Shahbaz Ali Shah from Department of Biotechnology and Genetic Engineering. I worked under the supervision of Dr. Qazi Muhammad Rafiq and Dr. Saadullah Khan. My topic of research was genetic analysis of sequence variants responsible for the achondroplasia and families of district cohort. These were the contents of my research. Coming to the topic, what is achondroplasia? Actually, achondroplasia is the malformation or genetic condition of our bones, in which there is absence of cartilage formation, due to which a condition occurs known as achondroplasia. In this condition, we can recognize a person by the symptoms of dwarfism, large head size with large prominent forehead, widened hands, short arms, broad legs, curved spine, etc. means all the malformations of the bone. It occurs about 1 in 30,000 live births and occurs in all races and in both sexes. Coming to the ROR2 gene, it is a receptor tyrosine kinase like orphan receptor 2 gene. This is coded by our ROR2 gene and it codes a protein tyrosine kinase and type 1 transmembrane protein. This is involved in the formation of chondrocytes and it is required for the formation of cartilage and growth plate development. Means it creates the formation of our bone. It contains about 228 nucleotides and consists of 9 exons and makes a protein that is of 943 amino acids. The current objectives of our study was to identify pathogenic variants responsible for achondroplasia and to establish genotype-phenotype correlation in families. So to accomplish the objectives, we first took the ethical approval from the Cust Research Ethical Committee, and then the families were localized from district cohort. Clinical investigations were carried out in order to confirm the achondroplasia phenotypes, and then the pedigree was drawn according to the information taken from the elderly person of the family, and then the blood samples were collected in EDTA containing tubes. The samples that is
Now perfect, you can start.
Okay. The blood samples were collected and the samples from affected individuals were subjected to whole exome sequencing. After the whole exome sequencing, which revealed heterozygous variant in ROR2 gene, the co-segregation of the variant in the family was confirmed using Sanger sequencing. And after the Sanger sequencing, different online available tools were assessed to check the pathogenicity of the ROR2 variant. So this is the pedigree of family A, showing different 3 generations of the family, and the circles show the normal female individuals, while the squares show the normal male individuals of the family. The filled circles and filled squares show the affected female individuals and affected male individuals of the family. And asterisk signs show that these members of the family participated in our study. So total 7 individuals were sampled from family A, and the whole exome sequencing revealed ROR2 genetic variant in this family. These were the clinical phenotypes of the family A, which are showing some facial features, showing enlarged head size, prominent forehead, a flat nasal bridge, short feet, dysmorphic jaw, trident hands, and disappropriate limbs. In ROR2 exon number 8, there was a mutation that is C.1189, where cytosine was replaced by thymine, and in which resulted in a termination of amino acid at position number 397, where arginine was present. Means early truncation or early stop codon, resulting in protein malformation. So these are the chromatograms of our family in which the unaffected individuals were homozygous, normal for the variant, while the affected individuals were heterozygous due to the dominant nature of the ROR2 gene. So this study highlights the utility of whole exome sequencing as a diagnostic tool for autosomal dominant disorders. In this, a nonsense mutation was recognized in ROR2 gene, that is C.1189C2T, and arginine 397 was converted to termination, which resulted in a protein which was not fully functional. And here in this family, we are for the very first time reporting the involvement of ROR2 gene in achondroplasia, as this gene has been previously known to be associated with only brachydactyly. So this is a novel research or novel conclusion from our result. These were the references of our information, all the information given here. Any question? This is all from my side.
Thank you so much, brother. It was really informative.
If you have any question, I can answer. No questions?
I think there are no questions. That
Assalamualaikum everyone. I am Hafiza Nida Shehzadi and I am a MS student at the University of Central Punjab, Lahore, Pakistan. I am glad that I got the opportunity to present at International Conference on Biological and Medical Sciences 2023. My research focus on reversal of available antibiotic resistance. In my MS, I examine assessment of efflux pump inhibition by flavonoids using berberine uptake assay against nosocomial pathogens. Nosocomial pathogens cause nosocomial infections, and nosocomial infection is the type of infections that a patient attends at the time of stay in the healthcare centers. A number of patients affected not only in the Pakistan but
you are not audible, Hafiza.
now am I audible?
Yes.
Okay, sir. And the alarming situation is that the 93.7% of isolates are resistant to third generations of cephalosporin. Further, carbapenem resistance bacteria show resistance to colistin, which is considered the last resort drug. So needs to develop the alternative strategies that become a development of new antibiotic or to break the resistance of available antibiotics. So I worked on breaking the resistance of available antibiotics by inhibit the efflux pump. So inhibition of these efflux pumps seems to be an attractive strategy at a time when the supplies of novel antibiotics are dwindling. The aim of this study was to inhibit the efflux pump and reverse the resistance of available antibiotics. So the first objective of this study was to assess the MIC of resistance antibiotic and flavonoids against obtained strain. The second objective was to evaluate the combination effects between the flavonoids and resistance antibiotic by checkerboard. The third one was to validate the efflux pump inhibition by berberine uptake assay, and the last one is in silico validation of wet lab analysis. The first step of methodology was to obtain bacterial strains and flavonoids, and in the second step was to identify the overly expressing efflux pump of bacterial strain by ethidium bromide agar cartwheel assay. And the next step is to determine the antibacterial activity through this diffusion assay, and then to determine the minimum inhibitory concentration of novel flavonoids through broth dilution method. And the fourth step is to determine the combination effects between flavonoids and resistance antibiotics through checkerboard. And the fifth step was to assess the inhibition of efflux pump by using berberine uptake assay. And the last step is in silico validation of wet lab analysis by using autotoc vena and pymol. The bacterial strains was obtained from Department of Basic and Applied Chemistry, University of Central Punjab, Lahore, Pakistan, and all type of characterization and antibiotic susceptibility testing was already done, and both strains were found to resistance to gentamicin. And the normal flavonoids at MURS013 and MURS014 was obtained in collaboration with Lahore University of Management Sciences. The activity of efflux pump was assessed by ethidium bromide agar cartwheel method. The results of the ethidium bromide cartwheel assay have proven the hyperactivity of efflux pumps as the strains were fluorescent at the highest concentration, that is 2.5 microgram per ml. Its mean ethidium bromide retained in the cells and its mean ethidium bromide retained in the cells and efflux pumps are active. Next, the agar disc diffusion assay was conducted using Muller-Hinton agar media for antibacterial activity of flavonoids. The inhibition zone greater than 7 mm was considered as positive. MURS013 alone has good inhibitory activity against E. coli C9 with 16 mm zone of inhibition, and MURS014 shows good inhibitory activity against E. coli 1841. But when combined with gentamicin, that found to resistant antibiotic showed best zone of inhibition against both strains. The minimum inhibitory concentration of flavonoids was also determined by broth dilution method. MURS013 displays lowest MIC 10.0 microgram per ml against E. coli 1841 and highest MIC was observed 12.0 microgram per ml against E. coli C9, and the MIC of gentamicin was also determined against the obtained strains. The slides move नहीं हो रही आगे. Can you hear me?
Yes.
Sir, slides next move नहीं हो रहा.
Now you can start.
So can I share the slides again because there is any problem? So my second objective was to investigate the combined effect of the extracts with antibiotics. MIC of both flavonoids and extracts MIC of both flavonoids and antibiotics was determined. Then based on the lower combined concentration where bacterial growth inhibition was observed, the fractional inhibitory concentration was observed that show additive interaction between flavonoids and antibiotics. And the third objective was to berberine uptake assay, and this assay was determined to assess the efflux pump inhibition by flavonoids.
You can see that the flavonoids reduce the MIC of berberine. The alone berberine shows MIC at 12.6 microgram per ml, but when combined with flavonoids shows MIC at 7.33 microgram per ml with 13 and 6.66 with 14. So, combination of efflux pump inhibitor with berberine enhance the antibacterial activity of berberine. So after that, the validation of antibiotic resistance reversal by flavonoids was determined by the same procedure that used for berberine uptake assay. And you can see that the MIC of gentamicin was reduced by both flavonoids, but shows good results with MURS 013. It means MURS 013 has good efflux pump inhibitory activity. And this was also validated through in silico analysis. Molecular docking was performed by using orthodopina. The ACR-AB-Tol-C with 13 shows lowest binding affinity, that is minus 9.8. Here is the 3D structures of interaction between ACR-AB-Tol-C with flavonoids that determined through FLIP, and the 2D structures between the flavonoids and ACR-AB-Tol-C through Discovery Studio. So developing a safer and effective plant-based API offer a promising solution to resolve drug resistance in pathogen. So our study linked with SDG goal 3, that for good health and well-being. This might be cost effective compared to developing new antibiotics. This breakthrough can provide it an alternative to combat growing drug resistance. This is all of my side. Thank you, everyone.
Thank you so much, Hafiza Nida. It was a beautiful presentation. Now I
Thank you.
request Sayyida Vardhani.
Yes, can you hear me?
Yes, you are audible.
Okay, I'm going to share my screen. Is the screen sharing?
Yes.
Okay. Hello, everyone, all the fellow presenters and guest speakers. My name is Sayyada Vardanur, and today I will be presenting our research on exploration of bacterial communities in Kera salt mine. This research has been performed by me, Sara Arshad, Sayyid Muhammad Abdullah, and our supervisor, Dr. Sarah Fazul. We are students from Capital University of Science and Technology. So first of all, we will be talking about the literature review, the research gap. I will show you some pictures from our Kara salt mine exploration, methodology, results, and discussion. And at the very end, I will leave some valuable insights about future recommendations. So our research began when our supervisor said that she wanted to work on halophiles, especially from Kara salt mine, which is the world's second largest salt mine. And according to literature review, we learned that in a salt mine, you can find wide variety of microorganisms from archaea, bacteria, and eukarya. Moreover, research revealed that till now, current research on Kera was only on Kera's soil, plants known as halophytes, and water, but none of it was on the salt, the most obvious place to look for microorganisms. So now we know that we're working with a gray area. Another gray area that we wanted to look into was the newly excavated salt mine within Kera. If you're not familiar, Kera is actively being mined for salt. And so we thought, okay, maybe we can find some evolutionary important microorganisms. So that decided our research gap, and we got started on our work. We went to Kera immediately after getting permission from Pakistan Mineral Development Corporation, who is currently dealing with Kera's mining and everything. And as soon as we got to Kera, we were surprised about
Can you guys hear me?
Yes, we can hear you.
Okay, unfortunately, I was knocked out of the meeting, but it's okay. So when we got to Kera Salt Mine, we saw that initially the salt was hard rock salt with black and brown color. As we went further, we saw white amorphous salt. And then we went into the museum area where we saw that there were many salt sculptures and artifacts. And there we saw that there were many different colors of salt. White, red, brown, and even shining peachy color, and they were literally glittering. And then after that, when we went to the newly excavated area, which will be on the left side of your screen, we saw that these salts were even more different because they had the color that Kira did not have. Another thing was that they were newly excavated, so there were least human contamination chances over here. So these were our 7 hotspot for salt collection. Then we get started on our methodology. Our methodology was very simple. We wanted to collect the salt, we wanted to perform the physicochemical properties, and then once we isolated the bacterial colonies, the main goal was to screen them for halophilic bacteria. One thing about halophilic bacteria is that they immediately need salt in the media, otherwise they will not grow. So among the 20 isolates that we got from the 7 salt samples, 19 of them passed the screening test when we grew them on 2% NaCl media. And then we went with the typical morphological characterization, biochemical characterization, and the next step was molecular characterization based on 16S rRNA sequencing. And then those 6 isolates were also taken further for biochemical testing, salt tolerance test, and antibiotic testing. Now, before I reveal the results to you, I want you to know that we have provided a key. The entrance salt was known as ETS, center salt, CTS, and museum salt, EDS 1, 2, and 3. And lastly, unexplored salt, which was the newly excavated area. Results of colony morphology revealed that these microorganisms were making diverse colonies. They were rhizoid, filamentous, they were off-white, they were opaque, they were yellow, they were orange, some were circles, some were like growing like roots of a plant. And then from microscopic analysis point of view, many of them were gram-positive. They had cocobacillus space shape or like rod shape. And the biochemical characterization reveals that all of them were catalase, oxidase, and sugar fermentation positive. But for the citrate and SIM test, they had a variety of results that I have listed in the table below. The next step was to understand where these microorganisms we were dealing with, what kind of salt concentration do they need? Because according to the literature review, you have 3 types of halophilic bacteria. So from like the ones that can tolerate 0.5% NaCl are those microorganisms that are known as slightly halophilic. And the ones that require 2 to 5% NaCl concentration are known as moderately halophilic. And lastly, the ones that need more than 5% NaCl concentration are extremely halophilic. So we grew our 6 isolates on 2% NaCl, 4% NaCl, and 6% NaCl. Surprisingly, they all grew on 2% and 4% NaCl, but not on 6% NaCl, which brought us to the conclusion that we are dealing with moderately halophilic microorganisms that require 2 to 4% NaCl concentration. Now, one would assume that how will we find any kind of antibiotic sensitivity or resistance even in these microorganisms that have never touched a human before? They're not in contact with any antibiotic, but we wanted to see it for ourselves. I mean, we presumed that we wouldn't find any resistance. Surprisingly, for the 3 antibiotics we used, erythromycin, ampicillin, penicillin, and chloramphenicol, all the isolates were sensitive to chloramphenicol, but they had a wide variety of resistances. And this was quite confusing because how did they get that resistance?
Excuse me, Saida. I'm really sorry to interrupt you, but can you kindly share your slides again?
The slides are not sharing. Okay, can you guys hear me?
Yes, you are audible.
Yeah, I'm so sorry. Someone just keeps removing me from the meeting. Okay, so from which slide did you guys miss out on the picture? Let me know and I can move back to that slide. Hello, can you guys hear me? Okay, please let me know from which slide should I restart again, because you guys told me that you cannot see the slides. Hello? Okay, so let's move on towards the results of antibiotic sensitivity results. As I was saying, that we did not presume that there would be any kind of resistance to these antibiotics, but we did see a wide variety of resistance towards erythromycin, ampicillin, penicillin, and chloramphenicol. But they were all sensitive to chloramphenicol, which was a great reassurance that, okay, we might have some kind of antibiotic that works on these helophiles. Upon getting our results for 16S RNA sequencing, we saw that molecular characterization revealed that we had 6 samples, and when we blasted their sequences on NCBI, we got their percentage similarity. Among them, UES1 and UES2, they both had 94% similarity, which was a really interesting contrast compared to the 100% similarities we got. And then all of them were mostly bacillus strains. Phylogenetic analysis confirmed that we were dealing with something that has been through extensive mutation over the course of time. For example, if you can see over here, the UES11 sample made its own clade. Similarly, UES22 sample was preparing its own clade as well. So there was something evolutionary happening here. Now, before I move on towards discussion, I would like to highlight that these microorganisms are currently under study for whole genome sequencing. So hopefully we will learn more about their phylogenetic and evolutionary relationship. Okay, so the next thing is the discussion. Now, of course, we were seeing that these bacillus species were the predominant ones that we were seeing. We now know that these current findings are related to the literature review, where Serra et al. Found that their species that they found from saline water in Cabra was similar to bacillus strains. And then another research on rhizosphere of helophytes was similar to bacillus strains as well, and they induced salt tolerance in plants. So the recovery of bacillus strains was showing that, okay, they have some ability to survive in the salt environment, survive as so much that they need salt to live on. The strain of bacillus like cuneiforms can be used to integrate any kind of probiotic, make bacitracin, or even to introduce salt tolerance into plants. But this was not the end. Like I said before, we're dealing with halophilic microorganisms. So they have diverse applications. They can be antimicrobial agents, they can produce enzymes, and moreover, they have antifungal and antibacterial properties. So this is not the end. This research has much more implications than we initially thought. So we're dealing with bacillus strains. The diversity is upon bacillus strains, and we now know that they can be used extensively. Another thing was that to explore the antibiotic sensitivity. How did they got that resistance? How are they? What is their genetic linkage towards that? At the end of that presentation, I would just like to say that Cabra itself remains unexplored. It has untapped potential in terms of microorganisms. I mean, PMDC, Pakistan Mineral Development Corporation, is focused on only extracting salt. They're not focused on any kind of research over there. Moreover, the newly excavated site in Cabra, it is waiting to be explored by researchers like us within Pakistan and international as well. And then another thing is that collaboration is a major role. I believe that Pakistan Mineral Development should have some collaboration with research companies within Pakistan and outside Pakistan. This would definitely be beneficial, especially to introduce salt tolerance plants into Balochistan, who is already dealing with drought and salinity. At the end, we would like to acknowledge our professor and supervisor, Dr. Sahar Fazl, the Dean of Biosciences and Bioinformatics Department at Capital University of Science and Technology. Without her insight, without her initial idea, we would not have made it so far, and we credit her for our huge success in finding 2 novel strains in the unexplored region in Kherba Salt Mine. That is the end of my presentation. If you have any questions, I am open to it.
Assalamu alaikum. My name is Nurlain, student of MPhil Zoology from Cohort, University of Science and Technology. The topic of my research is hymenolepiasis, a cross-sectional survey of associated risk factors and the circulating Hymenolapis diminuta. First of all, introduction. Hymenolapiasis is a widespread zoonotic disease caused by two tapeworms, Hymenolapis nana and Hymenolapis diminuta. It is present in the small intestine of rodent, especially mice and rats, and sometimes accidentally humans. It is a neglected tropical disease, and Hymenolapis diminuta is prevalent throughout the world, but it's only few hundred cases have been reported. Objectives: to evaluate the risk factors of Hymenolapiasis and to evaluate molecular characterization of this parasite targeting COX-1 gene. Material and methods. Material and method. First of all, sample collection. I collected the sample from different localities of district Banu. After that, I dissected the animals, means rats, and after that, I analyzed the intestine through the incision of sharp blades, and adult worms were present there. Fecal analysis was done through microscopy. After that, I extracted the DNA of these adult worms. Then after that, PCR amplification and at last gel electrophoresis. Results. It is the map of the study area. This study was conducted in district Banu. Different localities were selected, homes, for example, homes, shops, and different granaries where these rodents are found mostly and are abundant. Gender-wise comparison showed that this parasite was more prevalent in female as compared to male, but probability value was greater than 5%, which showed that the data is non-significant, means there is no relation between male and female and this parasite. Age-wise comparison showed that the parasite was more prevalent in mature rats as compared to young ones, and the probability value was significant. Season-wise comparison showed that the infection was higher in summer as compared to winter. Locality-wise comparison showed that this parasite was higher in poultry farms as compared to shops and homes. This is the PCR picture of amplification of COX-1 gene. The size of COX-1 gene was 699 base pairs. The conclusions of my research showed that the age play a significant role in the prevalence of the infection, means adult rats are most likely to be infected with this parasite. But while other factors such as seasonal variation, gender, localities are non-significant, they have no association. We concluded that the rat population at different localities is infected with this cystode parasite. And we also concluded that because of the close association of these rodents with the human habituation, and they can act as a source of zoonotic infection. Recommendations showed that extensive data collection across various districts is required. Rats control intervention should be needed, and further molecular analysis is required. Considering the zoonotic potential of this parasite, future research should focus on this parasite. These are the references. Thanks for your time.
Thank you so much, Noorul Ain.
I'm Amin Hassan. I'm an MPhil scholar at Kohart University of Science and Technology at Department of Biotechnology and Genetic Engineering. I would like to present my research work under the title of Molecular Analysis of Genetic Variants responsible for strabismus in families of district Kohart. Can you see my slides?
Your screen is visible, but not the slides.
Okay. Now,
Yes, ma'am.
so the topic for my presentation is strabismus. These are the outline of my study.
Ma'am, your slides are not visible.
Are they visible now? Hello?
Yes, ma'am, they are visible now.
Okay.
You can start.
Okay. Strabismus, also known as squint, is a common ocular alignment disorder. It is an alignment disorder, so our eyeball can be misaligned in various directions. The problem can be constant or it can be intermittent. Also, the severity can vary greatly. It results in amblyopia, blindness, and stereovision loss. The disorder can develop as a result of genetic factors or some environmental factors like smoking, advanced maternal age, injury, stroke, visual acuity, or other pathophysiological processes. Sets of 11 genes and 15 strabismus-related loci were recently compiled on a definitive genetic basis, out of which the 15 reported loci showed 8 candidate strabismus genes, which are given below. Can you hear me and the slides are visible?
Yes, you can continue.
Okay. So, and beside this, genes that are involved in a RAS-MAPK pathway also contribute to strabismus, and HOXB1 gene is associated with esotropia. Esotropia is a condition, it is a type of strabismus in which our eyeball is moved inward. Okay. The worldwide prevalence of strabismus shows that it is like of 2 to 6%, and it is likely to develop at any age, but it is usually developed during childhood. Also, in adulthood, it can frequently occur as a result of systemic diseases or mechanical damages like trauma or brain tumor, etc. The hypothesis for my current study was that in Pakistan, prevalence of these disorders are significant as there are different socioeconomic and traditional factors. So, strabismus being observed in different ethnic groups exhibit diverse phenotypes and genotypes, so that can contribute to an elevated incidence of visual impairment. Therefore, it is crucial to ascertain the specific variants responsible for strabismus, as a result of which this knowledge can facilitate many people and individuals about the timely diagnosis and genetic counseling of the disease. The objectives of my studies were to identify strabismus causing gene variants in certain families from the cohort region, as we know that we can say the gene are different at every region. So our main goal was to find the genetic variants from the cohort region in the individuals that were affected with strabismus, also to establish a causal relationship between the identified sequence variants and the phenotypic expression observed in the individuals within each family. Should I proceed? Should I proceed?
Yes, please.
Okay. The materials and method, or we can say the methodology for this research work was that first of all, we collected the blood sample from the both affected and healthy individuals. After that, the genomic DNA all the members of the families were extracted using standard phenol chloroform method. After that, the individual that were affected, their DNA were sent to whole exome sequencing. After that, PCR amplification was done. Before that, primer were designed, specific primer were designed. Then after PCR amplification, a segregation analysis was done using Sanger sequencing. What does the whole exome sequencing result shows? We will have a look at about the pedigree and the phenotype of my condition. This was the pedigree of my condition of my individuals of my family. There were 6 members, out of which only 4 members were sampled, and those one that are black, they show that they are the affected individual. And you can see the phenotype along with that pedigree. From the whole exome sequencing, it was concluded that there was a missense mutation at the chromosome number 11579, where nucleotide cytosine were changed into thiamine, and at protein level, propane was at the position 3860 was changed into leucocene. At the gene that were ADGRV1 gene. And after the whole exome sequencing, different bioinformatic tools were utilized to assess the pathogenic impact of variants on the protein like versome, CAD, mutation testers, and so on. For the in silico analysis, the mutation testing technique, mutation testing software were used, where it can tell you what type of a mutation was that and at which chromosome level, which cDNA level, and which genome level the mutation were found. From this study, we concluded that this can provide a valuable insight into the underlying genetic factors contributing to this condition. Also, advanced genetic techniques and thorough clinical assessment identified key genetic variants that are likely to play a significant role in the development of strabismus within these families. So, this was all about my presentation. These are the references. And thank you for giving me your precious time. If you have any question, you can ask.
Thank you so much, Ayman Hassan. If anyone have any questions, you can write in the chat box. I think there are no questions. Thank you so much, ma'am, Ayman Hassan. It was really insightful for us all. Now moving forward to ma'am Azka.
Can you hear my voice?
Yes, ma'am. You can continue.
Okay.
इससे भी अच्छी होती मैं इतना फ्रस्ट्रेट हुई हूँ ना सुबह से जैसी मैंने सोची थी ना वैसी बिल्कुल नहीं हुई मेरी प्रेजेंट
I have shared the slides. If these are visible, please give me a heads up.
Yes, they are visible. You can continue, ma'am.
All right. So I am Aska Habib from the University of Central Punjab, Lahore. I am doing MS in biochemistry and I have done my research. The title is Design and Synthesis of Novel 2-Nicotinic Acid-Based Siderophores to Promote Mineral Uptake and Enhance Plant Growth. Now we are starting with the introduction. Basically, what are siderophores and their function? Siderophores are the specialized molecules. They are produced by the microorganisms and their function is to, they are basically produced by the microorganisms and plants as well. And their function is to facilitate the uptake of iron from the environment. Now, these molecules, these siderophores, they have a high affinity for iron ions. and they allow them to chelate iron and transport it into the cells. Siderophores with high affinity for ferric ion, they are released out to create soluble ferric complexes, and these complexes will be absorbed by the organism. Now basically the siderophore iron absorption process begins with the chelation of ferric ion, resulting in the formation of iron siderophore complex. And we all know that iron is an important micronutrient. It is important for the growth of plants, and the iron content in plant is ultimately the main contributor of human health. Although it is present abundantly in Earth's crust. So I was saying that the main problem is that iron content in the environment, it's low for the plants to use it. So the main aim of my research project was to synthesize the chelators that will help in the development and growth of plants. How? By improving the intake of iron in iron limited conditions. So the objectives of my research was to synthesize the siderophores chemically, then the confirmation of the synthesized siderophores by some analytical techniques, and then to evaluate the siderophores for their capacity to increased mineral absorption, basically iron. Now moving on towards the materials and methods. Starting with the synthesis, basically, so the synthesis of siderophores have been done using chemically, and the chemical synthesis of siderophores have been done using 2 nicotinic acid metabolites. Now the characterization of my siderophores included determining their molecular weight, their chemical structures, and their functional groups that are responsible for their metal binding properties. And then for the characterization techniques, the techniques I am going to discuss have been used in the analysis, identification, and the characterization of my synthesized siderophores. The techniques included TLC. TLC was done to check the purity of my synthesized compound. Then FTIR, forward transformed infrared spectroscopy, was done. It gives us information related to the functional groups in our product. Then nuclear magnetic resonance helps us to interpret the structure of our novel synthetic compound. Then moving on towards the biological assessment. So in biological assessment, controlled plant experiments has been done that included collection of soil, germination of seeds, transferring the seeds into the trays, then managing the temperature and light throughout the experiment, observing the plants before any treatment, and then treating the plants with the synthesized compounds, and then documentation of the results, and finally ICBMS, which is basically inductive coupled plasma optical emission spectroscopy analysis to confirm the iron uptake. Now moving on towards the results. Here you can see that I have used 2 nicotinic acid and combined it with esters. So basically in my research, I took the 2 nicotinic acid, combined it with esters and amine. But in my presentation that I am showing, the slide that you are seeing, the reaction shows the reaction of 2 nicotinic acid with glycine methyl ester in a controlled set of experiment that included DMAP, EDC, then triethylamine, and I got my product. So basically, what was the purpose of using DCM? DCM was used to mix up everything. Then EDC was used for the activation of acid. DMAP, you can see DMAP. DMAP was used as a neutralizing agent, and triethylamine was used to balance the pH of the reaction mixture. Now I got my product. To determine the purity of the synthesized product, I used the TLC method. It helped me to identify any impurity. And after the synthesis of my siderophore, I went for their characterization that I discussed earlier, NMR and IR, FTIR, to determine the molecular weight, the functional group of the compound. Here you can see the FTIR analysis. So after the completion of my reaction, I went for FTIR, and the graph here clearly shows the formation of amide bonds, as we have peak at 3374 here. If you can see my mouse pointer, the peak here shows the formation of amide bond. Then the peak at 1745 shows the stretch of ester bond, and the peak at 1517, this one right here, it shows the carbon double bond carbon stretch of aromatic rings. And then at the end, we have carbonyl stretch of ether at 997. All these essential peaks shows the functional groups that are basically that are the part of my desired compound. Now moving on towards the biological assessment, the results from the biological assessment, you can see the visual documentation of phenotypic screening. The figure 1 shows the pots before any treatment in comparison to control and micronutrient control that was being treated with simple water. Then figure 2 shows the pots after the first treatment in comparison to control and micronutrient. Control was given simple water, micronutrient was given a solution of NPK, that was nitrogen, potassium and phosphorus. So you can see in the second figure, you can see a clear difference that in the height, a clear difference of height is visible in the second figure in comparison to control and micronutrient. And in the third figure, the picture is after the 6th treatment. The picture clearly shows a visible difference of controlled and my treated plant. To confirm the results that I got from the visual documentation, I went for to check the plant height and diameter first of all. Now the plant height and diameter, the graphs from the height and diameter shows a visible difference. There is an elevated enhanced height growth if we compare the treated plants with the control plants, and also enhanced diameter can be clearly seen in comparison to control. Moving on towards the chlorophyll quantification. To further check the results of the visual documentation, I did the chlorophyll quantification. The chlorophyll quantification clearly shows that we have an elevated level of chlorophyll in the pots that were treated with my synthesized siderophores. If I compare those to the pots, to the plants that were treated with simple water and micronutrient solution. And then moving on towards the ICBMS analysis, the bars here shows the presence of different minerals. Blue one shows the presence of iron, which was my main purpose, to enhance the iron uptake. And this graph, the fourth graph shows an enhanced iron uptake in the pots in the plants that were treated with the chemical 063 and 02729. It shows a clear difference if we compare it with controlled and micronutrient pots. Now moving on towards the discussion and conclusion. So basically the purpose of my research project was to enhance nutrient uptake, basically iron. Then to improve the crop yield, to support the agricultural and to reduce environmental impact of chemical fertilizers. And what I got at the end, I have observed increased plant growth in terms of increased plant height and increased diameter that were in the plants that were treated with siderophores. Then elevated levels of chlorophyll content have been seen in the pots that were treated with my synthesized siderophores, and of course enhanced mineral uptake. So that's all.
Can you kindly
sorry?
Thank you so much, ma'am. I was about to say that can you kindly conclude it?
Yes, that's all from my side.
Thank you so much, ma'am Aska. Now,
my name is Zahid Khan. I am student on IMPIL from Bahawalpur University. Today my topic is molecular characterizations of Anaplasma marginale in large ruminants of district Bajor. Introduction. My topic is related to anaplasmosis. What is anaplasmosis and what is the cause of anaplasmosis and how is anaplasmosis spread in these areas? Main cause of anaplasmosis is Anaplasma marginale. This is a parasite and transferred by ticks. Second, anaplasmosis cause many problems, human as well as animals, livestock, such as anemia, weight loss, miscarriage, severe cases of fatality. And this is the main symptom of what? This is the symptom of anaplasmosis. And all these diseases lead to many problem in animals. Their milk production was slowly and gradually decrease. My objective is that the molecular characterization of Anaplasma marginales, large ruminants of district buzzard. After that, scheme diagram or sketch palm. First of all, I collect blood, then gem sustain of forward for Anaplasma marginales to collect up Anaplasma marginal from blood. Then DNA extractions. After the DNA extraction is PCR, polymerase chain reactions, gel electrophoresis, DNA purification, and cycle sequencing of PCR. All of these steps I have done already for what? For Anaplasma marginal parasite. At the last, I will done protein analysis, draw cluster DAW, or second example is to blast this software for phylogenetic tree. After that, prevalence and district buzzard. From many areas, I have collected samples such as Salarzi, Barang, Khar, Uthman Khel, and overall regions around about 7.50 total ratio. After that, I done PCR, special PCR for this sample. I have selected two genes, MSP5 gene as well as 16S RNA. Around about, I collect 200 sample. 18 sample per PCR were positive, and result will come by PCR. Next, this is the phylogenetic tree of Anaplasma marginales, 16 sRNA. After that is MSP5 gene molecular surface proteins. This is a second genes I have select for Anaplasma marginales to make a phylogenetic tree as well as molecular detections. Conclusions. Conclusion is that for 16 sRNA as well as MSP5 genes of Anaplasma marginale, I collect first of sample, then by molecular characterization, I have done it. And as a result, I see means to analyze the characterization by sequencing, such as 16 sRNA and MSP5. Thank you. Any questions to ask me?
Thank you so much, Zahid Khan. Ma'am Sabha, you can start your presentation.
Okay, sir. So the slide is not visible. Okay. Assalamu alaikum, everyone. Bismillahirrahmanirrahim. My name is Sabah Hayat Mukhatab, and I have done my research work under the supervision of Dr. Shaukat Shuja. And the title of my research work is the synthesis of magnetic iron oxide nanoparticles for the photocatalytic degradation of methylene blue and methyl violet. So the content of my presentation include introduction, objectives, methodologies, experimental procedure, resultant discussion, and conclusion. So first of all, a short note on nanotechnology, that nanotechnology is the study and the manipulation of matter at incredibly smaller size, that is generally between from 1 to 100 nanometer. And nanotechnology is a rapid growing research discipline that investigate nanostructure with a recent focus on metallic and metallic oxide nanoparticles. That extensively studies the nanoparticles that are magnetic iron oxide nanoparticles, silver, gold, titanium, copper, and zinc nanoparticles. So nanotechnology is widely spread in different fields like physics, biochemistry, biology, life sciences, information technology, etc. So let's talk about the introduction about the nanoparticles. Like nanoparticles is a small particles that ranges between 1 to 100 nanometer in size. Undetectable by human eye, nanoparticles can exhibit significantly different physical and chemical property to their larger material counterparts. And the use of nanoparticles span across a wide variety of industries from healthcare, cosmetic to the environmental preservation and air purification. So next one is the introduction that nanoparticles are mainly classified into two types, inorganic and organic nanoparticles. The inorganic nanoparticles include metal and metal oxide nanoparticles, while the organic nanoparticles include quaternary ammonium compounds, cationic, quaternary polyelectrolyte, etc. So let's talk about the objectives. So the objectives of my research work is to synthesize and characterize magnetic iron oxide nanoparticles using nanoroff Ricnia leaves extract. And the second one is to assess the potential of synthesized magnetic iron oxide nanoparticles for the degradation of methylene blue and methyl violet. So next one is the methodology. So the methodology include the preparation of plant extract, synthesis of magnetic iron oxide nanoparticles, characterization techniques that include FDR spectroscopy, SEM, ADX, XRD application like photocatalytic degradation of methylene blue and methyl violet dye. So the experimental procedure that include, first of all, the nanoroff Ricnia plant leaves is taken. After that, 10 gram of leaves is aerated into 100 ml distilled water. The extract is formed. After that, two different salts are used, that is the ferric chloride and the ferrous sulfate. So 0.1 molar ferric chloride solution and 0.1 molar ferrous sulfate solution are added and 2:1. After that, it is centrifuged, and last the nanoparticles are synthesized by centrifugation, and it is dried and stored for the characterization techniques that include FTAR, ADX, SEM, and XRD. So about the result and discussion that include characterization technique that I explained earlier. So the first one is the FTR analysis of plant extract. So the figure 1 shows the FTR spectrum of plant leaves extract that shows different kind of bands. You can see peaks that is hydroxyl, carbonyl, CN, NH, and CO stretching. So the next one is the FTR analysis of magnetic iron oxide nanoparticles. So the main peaks are labeled here, that is the sp3ch stretch, and one of the important peak, that is FEO. That is from this peak, it is clear and evident that the magnetic iron oxide nanoparticle is synthesized by using plant leaves extract. And the next slide is about the SIM images. So here the SIM images at a different magnification. So that shows the nanoparticle are spherical in shape, and the size of nanoparticle ranges from 1 to 15 nanometers. So this is the XRD for magnetic iron oxide nanoparticles. Here the different peaks are given, and you can show from these peaks the crystallinity of the nanoparticles. So by using Schur equation, the crystallized size of nanoparticle are calculated, that is 7.31 nanometer. And the next one is the EDX analysis. So figure 4 show the EDX spectrum of magnetic iron oxide nanoparticles. So from the table and as well as from the peaks, we can conclude and we can say that the iron is present in a huge amount, that is 66.98 by weight and 42.55 by atom. So the synthesized magnetic iron oxide nanoparticles are further used for the application purposes, like for the photocatalytic degradation of methylene blue and methyl violet dye. So first of all, we have synthesized different. So we have to form different concentration of methylene blue dye for the purpose of degradation. So this is the blank values of methylene blue, and the regulation graph is also given here. So from the next slide, we can show the percentage degradation of methylene blue dye as well as the catalytic degradation of methylene blue. So the contact time, next one is the contact time. Methylene blue dye was degraded in the presence of magnetic iron oxide nanoparticle, which act as a photocatalyst. So methylene blue dye was degraded by adding 0.1 milligram of the catalyst. After that, the maximum absorption was checked by a different time interval from 0 to 120 minutes. So the change in color was observed from blue to colorless solution due to the presence of catalyst, and after that 120 minutes the dye was completely degraded up to 85%. So this is the contact time parameter that from this graph you can show from 0 minute up to 120 minutes. So at 120 minutes the dye was completely degraded and it acquired a flat pattern that can be shown in the graph. So this is the degradation of
Ma'am, can
methylene
you
blue at
kindly
different pH
conclude?
levels. Yes.
Excuse me, ma'am. Can you kindly conclude?
Okay. It's just a 2 slide and I concluded. Okay. So this one is the photocatalytic degradation of methyl volet dye, and after that it is the catalytic degradation of methyl volet, and the percentage degradation of methyl volet is also given. So last one is the conclusion. So magnetic iron oxide nanoparticle was prepared by using nanorough Frignal use extract, which is very cheap and easily available. So the synthesis of magnetic iron oxide nanoparticle was confirmed by color change of solution from brown to black. And also due to the magnetic nature of nanoparticle, and the FTR and XRD result further support the formation of nanoparticles, and methylene blue and methyl volet dye was degraded up to 90%. So these are the references of my research work, and I'm truly grateful and thankful to my respected and dedicated supervisor, Dr. Shaukat Shuja. And this is all about my presentation. So any kind of question you can ask.
Thank you.
Bismillahirrahmanirrahim. Assalamu alaikum. I'm Zainab Bano, MPhil scholar from Department of Zoology, Bahawalpur University of Science and Technology, Bahawalpur. Today, the topic of my presentation is molecular characterization and phylogenetic analysis of Anopheles stephensii. The outlines of my presentation is introduction, rationale, aims and objective, methodology, results, conclusion, references. Introduction. Mosquito belong to family Culicidae containing approximately 35,000 species. Mosquitoes are vector for number of diseases like malaria, dengue, yellow fever, etc., causing morbidity and mortality worldwide. Half of the world's population is at risk of being infected with at least one type of vector-borne disease. Malaria is endemic in Pakistan. According to World Health Organization 2021, Pakistan is one of the 7 countries in Eastern Mediterranean region that count for 98% of total malaria burden in the region. In 2021, about 2.6 million suspected cases of malaria were reported in Pakistan. In 2022, over 3.4 million suspected cases of malaria were reported. Malaria is caused by the bite of female Anopheles mosquito. Anopheles stephensii is an Asian malaria vector causing urban malaria in Pakistan. Anopheles stephensii has 3 biological forms. Number 1, mesorensis, intermediate, and type. Anopheles stephensii has expanded to new geographical localities, posing a threat to the elimination of urban malaria. It was first detected in Djibouti in 2021. Since then, it has been reported in Ethiopia, Sudan, Somalia, Horn of Africa, and most recently, Yemen. Rationale. The selected area is a variation of mosquito due to the junction of different districts as well as the presence of stagnant water and pond water. Mosquito phenistic study have been carried out in different parts of KP, but molecular study has not been done before. The study conducted in the area for the first time. Objectives. The objective of my study is to investigate the diversity of mosquito in Pakistan and to assess the molecular characterization of Anopheles stephensii by using odorin binding protein 1 gene. Methodology. Mosquitoes collection is performed indoor. Indoor collection is done in water bodies and plant and vegetation by using hand net, aspirator, and light trap. Indoor collection was made in homes and animal shed by using spray catch method. The collected mosquitoes were preserved in 80% ethyl alcohol and identified under stereo microscope by using taxonomic key, the fauna of British India, Anopheles stephensis were separated and DNA were extracted, PCR was performed, and phylogenetic analysis was done. This slide shows the map of KP showing the cohort region. On the right side, in cohort region, the 4 villages which were selected for the survey of mosquito collection are given. These 4 villages are Ustarzai, Jarma, Lachi, Togbala. Data analysis. For data analysis, relative abundance was calculated by using the formula given in the slide. Relative abundance is equal to n divided by total number of species multiplied by 100. If the relative abundance is more than 10%, the species was tagged as dominant species. If the relative abundance is between 3 to 10%, the species was called subdominant species. And if the relative abundance is less than 3%, the species was called satellite. Shannon-Weaver index. It was used to find out the diversity of mosquito. Shannon index Weaver's formula is given in the slide. Species evenness was calculated for each mosquito species in 4 localities by using the following formula. Species richness was calculated by Margalef index given in the slide. Margalef index was used for species index in all the 4 villages. Results. The result of my study is that 4 genera were collected. 3 genera were collected. Culex was the most dominant genera, which was 76%, followed by Anopheles, 22%, and Aedes, which was the least dominant genera, was 2%. This slide will show the 7 species belonging to 3 genera and showing the relative abundance in each villages. The 7 species are Culex quinquefasciatus, Culex mimeticus, Culex thaleri, Anopheles stephensi, Anopheles culicifasi, Anopheles annularis, Aedes aegypti. The highest number of mosquito was collected in Ustarzai 3070, while the least number of species collected was in Togbala 1860. This slide shows the relative abundance of mosquito collected from cohort. Culex quinquefasciatus, Culex mimeticus, Culex thaleri, Anopheles stephensi was tagged as dominant species. Anopheles culicifasi and Anopheles annularis was called subdominant species, while Aedes aegypti was termed as satellite. This slide shows the picture of mosquito collected from district cohort. This slide shows the number and sex ratios of mosquito collected from district cohort. The highest number goes to Culex quinquefasciatus, 4,280. The ratio of female to male is 2.39. For Culex mimeticus, the total collection was 2,390 and female to male ratio is 1.87. For Culex thaleri, the total number collected was 1,490 and female to male ratio was 1.98. Anopheles stephensi, the total number collected is 1,170 and female to male ratio is 2.77. Anopheles culicifasi, the total number collected is 850, female to male ratio is 1.65. Anopheles annularis, total number collected is 300 and female to male ratio is 3.66. Aedes aegypti, the total number collected is 260, while female to male ratio is 2.25. Species diversity of mosquito collected in 4 villages is given in this table. The highest species diversity was recorded in Jarma, which is 1.74, followed by Lachi 1.61. The least species diversity was recorded in Togbala 1.34. Highest species richness was recorded in Lachi 1.60, 1.06, while least was recorded in Togbala 0.95. Species evenness highest was recorded in Jarma 0.89, while the lowest was recorded in Togbala 0.69. This slide shows the monthly species composition of mosquito in different localities of Kohat. The highest number of mosquitoes were recorded in June. In Jarma, it was 1,380. In Ustarzai, it was 1,320. In Lachi, it was 1,310. And Togbala, it was 1,250. While least number of collection was recorded in January. In Jarma, in January, it was 40. In Ustarzai, it was 30. In Lachi, it was 20. And in Togbala, it was 10. Molecular characterization of Anopheles stephensi. From the collected mosquito, Anopheles stephensi was separated and molecular characterization was performed. DNA extraction. The DNA of Anopheles stephensi was extracted by using Kygene DNA mini kit, following the standard procedure of manufacturers. The extracted DNA was stored below -4 degrees centigrade. PCR. Amplification of Anopheles stephensi odorant binding protein 1 fragment was performed by using forward and reverse primer. The size of primer was 845 base pair. Thermal profile used for the PCR was denaturation temperature was 95 degrees centigrade. Annealing temperature was 60 degrees centigrade and extension temperature was 72 degrees centigrade. 30 cycles were repeated. This picture shows the PCR amplification of 485 base pair fragment of Anopheles stephensi. Phylogenetic analysis. PCR amplified product was sequenced commercially. The final sequence were aligned with cluster W with homologous sequence downloaded from GenBank, and phylogenetic analysis was constructed using distant neighbor joining method based on Temura-Nye model in MEGA 11. 120 base pair fragment of Anopheles stephensi odorant binding protein 1 intron 1 region was used for analysis. The comparison of sequence with the representative sequence was used for making the phylogenetic analysis, which shows that Kohat specimen was all type form. Anopheles stephensi type form was the prevalent biological form in Kohat specimen. This picture shows the phylogenetic analysis of Anopheles stephensi odorant binding protein intron 1 region, which shows that all the specimen belongs to type region. The bootstrap value was 1000. Conclusion. Culex was the dominant genera, followed by Anopheles, and Aedes was the least dominant genera. Density of mosquito was lowest in January and highest in June. Culex quinquefasciatus and Culex mimeticus was the most commonly occurring species found throughout the study. Anopheles stephensi form was prevalent biological form in Kohat specimen. Anopheles stephensi odorant binding protein intron 1 sequence could be introduced as a molecular marker for the detection of 3 biological forms. References, acknowledgement. I'm highly thankful to my supervisor, Professor Dr. Shahid Niaz Khan, and all the staff members of Department of Zoology, Kohat University of Science and Technology. Thank you.
Hi, hello, Assalamu alaikum everyone. My name is Muhammad Akeem. I'm a student of MS Microbiology, Kohat University of Science and Technology. So I present my research work on the topic of molecular characterization of bigomovirus complex impacting capsicum enema and Malvestrum chromandelium plant and Kohat district. So I start from the outline. Here is the outline of my presentation, including introduction, material and method, results, and references. Here is the introduction of my presentation. So I start from the bigomovirus. Bigomovirus is the one of most devastating genus of the family of Zemeniviridae, having a small circular single-stranded DNA genome. A bigomovirus genome is either monopartate designated as a DNA or bipartate designated as a DNA and DNA-B. DNA A contains 6 overlapping open reading frames, ORFs, 4 ORFs on the complementary strand and 2 on the virion strand. Large intergenic region is a period between complementary strand and virion. Bigomovirus are usually associated with a satellite molecule called alpha and beta molecule, which are approximately half in size of their helper virus. Alpha and beta satellite have 1 RF each, encode for replication associated protein and beta C1 protein. So bigomovirus only transfer by the whitefly. They have a much bigger problem for a wide range of cultivated and non-cultivated plant, including cash crop, ornamental plant, vegetable, feed, etc. They cause significant economic loss across the globe. It includes a lot of crop like cotton, one of the most economical crop in Pakistan. Introduction is continuous, so I start from the plants which I choose from for my research, my MPhil research. So first of all, I main focus on chili. Chili belong from the family of Solanaceae. Chili are widely used as a vegetable as well as an herbal, pharmaceutical, and cosmetics. Chili is a significant crop, cash crop in Pakistan, particularly in Sindh, followed by Punjab, Balochistan, and Khyber Pakhtunkhwa. The grain is grown on the area of 64.8 thousand hectare and the product 142.1 thousand tons. The production is affected by various pathogens, including bigomovirus. So the next plant I choose for my research, it's Malvestrum. Malvestrum belong to the Malvaceae family, and it's frequently in warmer region. Malvestrum is weed survive as a hub for multiple bigomovirus. It's a big problem, providing a chance for evolution of new variant strain and species of bigomovirus. So the next slide is all about material and method. First step include in material and method is sample collection. I collect symptomatic sample from various field and a district cohort, and then I brought for further process in a lab known as a PVL plant virology lab. After that, I proceed very carefully the sample for DNA extraction. For DNA extraction, I follow a standard method, CTAB method, which is standard for a plant, which is standard for plant extraction of plant DNA. So after that, I verify the DNA on 1% agarose gel for before using for PCR. After that, when DNA is extract, I further the next step include is molecular identification of picomovirus by using a polymerase chain reaction. For application process, I used many primer, and one of the selective primer is beta 01 and beta 02 for amplify DNA 1 and DNA 2. Beta 01, basically beta 01 and beta 02 is a universal primer for the identification of picomovirus. Either it's DNA A or DNA B. After a PCR product, after application of PCR product, will purified, clone, and sequence commercially. Next step include bioinformatic analysis. After sequencing, I use bioinformatic tools to make the data graph, etc. I use a lot of software, including STD species demarcation tools and recombinant RDP recombinant detection program and plotcon, etc. Results. This picture basically it's a picture of 1% agarose gel. Basically, lane 1 show 1 kb ladder, and others lanes all about the DNA of chili and malvestrum, which I extract and isolate from the different symptomatic sample of chili and malvestrum. Result of PCR product. These both pictures confirm the application of picomovirus. In both picture, lane 1, I used 1 kb ladder for the confirmation of my desired product. I used 1 kb ladder for the confirmation, and I mentioned in material and method I used the universal primer for the amplification of picomovirus, which is known as a beta 01 and beta 02. The size of beta 02 and beta 01 is 1350 base pair. So these pictures confirm the virus in both sample. References. It's the references of my presentation. Thank you very much for listening to me.
Assalamu alaikum, sir. My name is Shafqatullah, MPhil Scholar of Zoology, Department of Zoology from Kuhat University of Science and Technology. My supervisor, my first supervisor is Dr. Abdul Majid, Department of Zoology, and also the second one is the Dr. Muhammad Qasim, Department of Microbiology. Today I am going to present the molecular detection and phylogeny of rickettsia species circulating in ticks and calls of district Banno. My presentation outline is start from the introductions that rickettsia is a genus of gram-negative non-spore forming aerobic bacteria that are obligate intracellular parasite of eukaryotic cells, which is responsible for a variety of disease in humans and animals, including which is caused the rickettsioses and the Rocky Mountain spotted fever, typhus, and Q fever. The emergence of antibiotic resistance has led to an increase in zoonotic infection, which can cause significant morbidity and mortality. Rickettsia spread through a mammalian host bite from ticks, mites, and the contaminated droppings of infected fleas and lice. By how we can detect that which types of the symptoms of rickettsial infection can include in the patient, that it includes the fever, headache, muscle pain, and rashes. So Pakistan is a large agriculture country which have contribute in agriculture industry employees of 43.4% and in the 20.9% of the total GDP in Pakistan. So the objective of my presentation is According to the rationales or the objectives of my presentation, according to the best of our knowledge till date, here there is no published work on the molecular epidemiology of rickettsia species which are detected in district Borno. So the current study was carried out to detect the rickettsia species in ticks and livestock of district Borno, which will helpful for the designing control strategies of rickettsia disease of livestock. So the objectives of my presentation is to detect rickettsia species in ticks and calls of district Borno, and the second objective of my presentation is to detect the phylogeny or analyze the phylogeny of rickettsia species from district Borno. So this is the graph representations of the conference, which are detected different types of rickettsia in different areas of the district Borno, area wise prevalence. So Dumel, Nizambazar township, Kaki, Nuranis, Sadehel, Sarkihel, total you have 13.33%. So there are two areas in the district Borno, the Sadehel and Sarkihel, which do not have any contributions of the health facilities of the district Borno. So they are very prolonged from these facilities. Also they have the graph representation to have a large amount of the affected animals in this area. After this, I represent the second slides of the presentation in the month-wise prevalence. Month-wise prevalence. So the ticks are also active in June, July, August, September, October, November. Overall, the 2 months, October and November. In these months, the ticks are highly active in these months. So the 22.920 and 20.41 is also the affected areas which are prevalence in these areas of the ticks. The gender wise prevalence, this is also affect the male and the female gender. This is the molecular phylogenetic analysis which are designed by the maximum likelihood method. This is the samples of our study 4, 1 and 5, 3, 6 and 2, which have a great similarities with these rickettsia species. And also this rickettsia species is designing for our study to have maximum similarities with the different countries where presents the rickettsia species, rickettsia rickettsii, rickettsia picikoi, rickettsia siberica, rickettsia species honai, rickettsia japonica. This is also the different countries rickettsia species. With this, if we compares of my samples with these different countries rickettsia, there are some similarities which are responsible for the detection of rickettsia species in the district Borno. This is the molecular phylogenetic tree which are on the country basis, and this is the gel picture which are represents after the PCR analysis. And this is the conclusion of the and recommendation of my study, and this is the acknowledgement of my presentation. So thank you, sir. If any questions.
Assalamu alaikum. I am Sara Gul, a student of Chemistry, Guwahati University of Science and Technology. My first supervisor name is Sir Shaukat Shuja, and my second supervisor name is Sir Taslim Hussain. And my topic of presentation is green synthesis of zinc oxide and copper doped zinc oxide photocatalyst for degradation of methylene blue. 21st century biggest challenge is giving people access to clean water. In this regard, different technologies are developed, but in the era of nanotechnology, metal nanoparticles have gained importance over traditional methods. So in this research, copper doped zinc oxide nanoparticles were prepared to degrade dicontaminants in the water. So different doped concentration of used to synthesize zinc oxide nanoparticles via green method, and the sample characterization was done by UV, XRD, FTIR, EDX, and methylene blue is used as in the photocatalytic investigation of nanoparticles. The activity of 3% and 5% samples were superior, so the synthesized samples were eco-friendly and can be used for the treatment of dyes. Introduction. Due to growth of dyeing industries in recent years, dyes are generated in large quantities, and most of these dyes are poisonous in nature. Also, scientists use various physiochemical processes to remove these dyes from water that have some disadvantages. The use of biosynthesized semiconductors gained a lot of importance to remove waste from the water. Among semiconductors nanoparticles, zinc oxide attracted research attention due to many properties over other nanoparticles. And their photocatalytic activity can be increased by doping with copper, so that copper reduce their band gap and also increase their photocatalytic activity. In this regard, also the plant mediated technique is used, and the plant choice was the Nanorophrycnia to produce the copper doped zinc oxide nanoparticles. The well-known dimethylene blue has used in many industries, and it causes many health issues. So it will be used to analyze photocatalytic activities of zinc oxide nanoparticles. Objectives: synthesize zinc oxide and copper doped zinc oxide nanoparticles, characterize them, and also to photocatalytic degradation potential of copper doped zinc oxide nanoparticles. Synthesis of extract. In this regard, plant extract was prepared. Firstly, the coarse powder of Nanorophrycnia was wetted, and 25 gram was dissolved in 400 ml distilled water, and then it is heated 80 degrees centigrade for 30 minutes. Then the filtrate was it was filtered, and the filtrate was stored for further use. Synthesis of zinc oxide nanoparticles. Zinc oxide nanoparticles were prepared by using 100 ml plant extract, and then 200 ml distilled water was added. After that, 300 ml of 10 millimolar zinc nitrate hexahydrate was poured into the extract, and it was constantly heated at 60 degrees centigrade and stirred. Also with that, sodium hydroxide was added dropwise until the pH of the solution was reached to 10. Then mixture was heated constantly at 60 degrees centigrade until a yellow paste is formed. The paste was calcinated at 600 degrees centigrade for 2 hours, and after that the pale white powder was obtained, which is stored for further use. Synthesis of copper doped zinc oxide. Copper doped zinc oxide was prepared by the same method as described above, but with the addition of copper chloride salt dropwise. Different copper concentration were used: 1%, 3%, and 5%, weight by weight copper doped zinc oxide was prepared. Characterization. UV was used to confirm the formation of zinc oxide nanoparticles, and FTIR was used to confirm the function of functional group that are present in the produced sample. TEM was used to verify the crystal structure of the synthesized sample, and XRD was used to check the crystallinities of the sample, and ADX was used to check the doping concentration on the sample. Application: photocatalytic degradation of dye. In order to measure the photocatalytic activity of copper doped zinc oxide nanoparticles, 4 mg of catalyst was introduced into the 20 ml of methylene blue of 10 ppm solution, and after 30 minutes of sonication, it was exposed to light, and throughout the experiment, the photocatalytic activity was measured. Dye degradation was optimized at dye concentration, pH, time, and temperature. Conclusion. In this study, copper doped zinc oxide nanoparticles were prepared through green synthesis and their efficiency were evaluated for photocatalytic degradation. Zinc oxide copper 5% were most efficient catalysts among other and can be used for the degradation of methylene blue. And next is references. Thank you.
Me, Mujahid Rahim, Supervisor Dr. Kalimullah, and Co-Supervisor Dr. Abdur Rahman. I am student of Cohort University of Science and Technology. My discipline is MPhil Zoology. Today my presentation topic is impacts of diazinon on hematological and histopathological indices of Labiurheta in district cohort. The outline of my presentation is introduction, objectives, materials and methods, results and discussion, summary and conclusion, recommendation, references and acknowledgement. So first I introduce what is diazonane. Diazonane is a colorless to dark brown liquid. Diazonane is widely used in insecticides to control pests in different countries. Based on report of the Bulletin of Agriculture Ministry 2008, the annual consumption of diazonane in Iran is estimated to be 3775 tons. Diazonane is synthetic organophosphate which is primarily used in agricultural purposes. Diazonane is after the treatment with the diazonane to control pest, non-targeted animal is also exposed to the pesticides. So it is drawn in the different rivers and with the different concentrations. Average half-life of diazonane is 29.9 to 34.8 days respectively in soils. Due to this half-life cycles, the diazonane due to housefly activities, agriculture, run off directly and discharge to stream, ponds and other aquatic conditions. So it affects aquatic organism, particularly fish. Labiurheta belongs to order Cypriniformes, family Cyprinidae. It is native to the water system Pakistan, Bangladesh, India. It is the most common fish of Pakistan market and commonly consumed in Pakistan. Because Labiurheta grow easily and it is available in our market of Pakistan. Due to this, it is the most common fish of Pakistan. Effect of diazinon on the hematological parameters of fish. Diazinon affected the hematological parameters of rainbow trout. It reduced the RBC, WBC, PCV, HB and neutrophil level. Diazinon also affect the hematological parameter in Carpio. It reduced the PCV, RBC, HB, lymphocyte, leukocyte, and non-specific immunity, and also show diazinon also show effect on the hematological parameter of different fishes. Effect of diazinon on the histopathological parameter of fish. Diazinon show negative effect on liver in case of rainbow trout. Histopathological damage were also seen in Kipota. The result of histopathological analysis revealed that diazinon export cause abusive injury in corp gills. Rationale of my study is that previously has no research work done on the impact of diazinon on hematological and histopathological indices of Labiurheta in district cohort. No study have been conducted that assessed the impact of diazinon on the hematological and histopathological indices of Labiurheta in district cohort. Objective of my presentation and my research is that to find out the effect of diazinon on the hematological parameters of Labiorheta or Rahufish. To find out the effect of diazinon on the gills and liver histopathological parameters of Labiorheta. The second one we can see, in other words, to find out the different spots and the gills and livers of Labiorheta which is affected by the diazinon. Materials and methods. Material and method. First, we can collect the fish from different local forms of district cohort. Then we can acclimatize in the laboratory of Department Zoology, Cohort University of Science and Technology for 15 days. After acclimatization, we can collect the blood from the caudal fin of the fish through syringe. This blood is collected, is saved is placed in dropped into the EDTA tubes in order that the blood does not clot. After this, we can carry this blood into laboratory for the blood analysis. In the laboratory, we can put the EDTA tube under the hematological analysis. From this machine, we can conclude that we can take the different hematological parameter test. And histopathological analysis of gills and liver. First of all, we can take the fish from different local form of district cohort, and then we can acclimatize them in the laboratory of Department of Zoology, Cohort University of Science and Technology. After acclimatization, after 28 days, we can dissect the fish. From the dissect, we can collect the tissue of liver and gills from the dissected fish. So for the further processing, we can carry these tissue in the laboratory of KMU Khyber Medical University. So the first step of histopathological is tissue processing. After that, impedance station. After it, microton. After that, preparation slides. After that, staining of the slide, and the last is microscopy, which is I use the microscope Olympus. So it is the method of the histopathological and hematological analysis. So my presentation and my research, the hematological analysis of control and affected diazinon affected fish were seen in the table in the slides that we can get from that. So we can place 28 days of fish in the laboratory. After 28 days, we can collect the blood from this. When we take the blood from this, the hematological parameter like RBC, WBC, HB, HT, MCV, it is significantly decreased in the both affected diazinon groups. 2 microgram per liter groups as well as the 5 microgram per liter group show a significant decrease in the hematological parameter of blood like RBC, WBC, HB, HT and MCV. The control group do not have any decrease in these parameters. Histopathological analysis of liver. Representative or the below image show liver section of fish seen under the Olympus microscope with 10x from control and both diazinon affected group. After 28 days, we can I can dissect the fish. After the dissection, I collect the tissue of liver and gills. When we check these tissue and the Olympus microscope. From the Olympus microscope, I concluded that control group do not show any morphological change in the liver. It show a normal liver tissue with the hexagonal haptic cells, while the both affected means that 2 microliter per liter and 5 microgram per liter, 2 microgram per liter diazinon and 5 microgram per liter diazinon show a significant morphological change, and the liver histopatology of labiorheta like the localization of cytoplasm, necrosis of the liver cell, etc. Histopathological analysis of gills. The representative image shows the gill section of labiorheta. Labiorheta subjected for 28 days, and they are two different doses of the were taken. So after 28 days, control group fish showing normal gills, while the fish which is exposed to 2 microgram per liter showing epithelial hyperplasia, while the fish which is subjected to 5 microgram per liter display epithelial rising, epithelial hyperplasia, and fusion of secondary lamella. Summary and conclusion of my presentation below. I concluded that the significant decrease in RBC, WBC, HB, HT, and SAB, and also indicate that severe morphological change in the liver tissue, lost heptic cells, lost polygonal structure, localization of cell cytoplasm, cloudy swelling, and liver tissue and nucleus atrophy. Control group of labiorheta has the normal structure of liver with a hexagonal haptic cells, while the affected show morphological change in liver tissues. It is indicated that severe gill damage are also occur in the infected fish, including epithelial hyperplasia, epithelial risings, epithelial hyperplasia, fusion of secondary lamella, and excessive mucus secretion, while the control group of labiorheta show normal gill structure. They have no morphological change. Recommendation of my presentation: to perform more research to test whether these effects are determinal to other fish species in the field. Number 2, the alternative to synthetic chemical pesticides should be encouraged for pest suppression in agriculture. The government should emphasize research and extension activities related to IPM program for minimizing the use of chemical pesticides. These are the references of my research that I can help from these papers. In acknowledgement of my presentation, I am very thankful to Dr. Kalimullah, Assistant Professor, Department of Zoology, Guwahati University of Science and Technology, Dr. Abdul Rahman, Assistant Professor, Department of Microbiology, Guwahati University of Science and Technology, Dr. Said Ishtiaq Anjam, Chairman, Department of Zoology, Guwahati University of Science and Technology, Mr. Muhammad Rauf Afridi, Lecturer in Department of Zoology, Guwahati University of Science and Technology, Dr. Muhammad Ishtiaq, Assistant Professor in Beijing University, China, Dr. Naveed Sharif, Assistant Professor in KMU, Muhammad Rahim Khan, DMLT, Sarid Khan, DMLT, and all my research in lab mates, especially Karimullah, Hidayatullah, and Muhammad Zishan. I am very thankful to CURST and Higher Education Commission for giving me opportunity of my MPhil study. Any suggestion? Thanks.
Bismillah ar-Rahman ar-Rahim. My name is Zahid Khan. Today my topic is molecular characterization of Anaplasma marginale in large ruminants of district Bajor. This is my title of the research. Introduction. Anaplasmosis is caused by Anaplasma marginales, mainly target vertebrate and invertebrate organism and their relations with Anaplasmataceae family. They mainly target vacuoles membrane. Acute anaplasmosis causes severe fevers, hemolytic anemia, weight loss, miscarriage, as well as in severe promiscuity, and affect the milk production and meat production. The relations of Anaplasma marginalis to genus Anaplasma, and they transfer by vectors such as ticks. My objective is molecular characterization of Anaplasma marginale and large ruminant of district Bajor. First is collect blood, then gene sustaining. After gene sustaining, DNA extraction, PCR, gel electrophoresis, DNA purifications, and blast of these sequence to make a phylogenetic tree. This is the percentage of male and female. I collect blood from these organisms and the suspected cases. This is the picture of gel. Clearly represent that MSP5 genes banned by gel I have run. This is the example of 16S RNA gene, clearly represent that 16S RNA I targeted, and by gel come positive. This is the phylogenetic tree of 16S RNA and relate with many countries: USA, Australia, Thailand, China, etc., India as well as India. My topic is related by sequence through these countries. This is MSP5 genes. MSP5 genes, I selected MSP5 genes. This is a molecular characterization and the sequence related with many countries: India, Egypt, Thailand, Brazil, China, etc. Conclusions. My topic is molecular characterization and make a phylogenetic tree, but I select to 16S RNA and MSP5 gene, and I find out these genes molecular characterize DNA extractions, PCR, then gene electroporation. At last, I make a phylogenetic tree of these two genes and find out correlate with other countries as well as Pakistan.
Okay. The topic of my presentation is Caralluma tuberculata mediated synthesis of copper oxide and reduced graphene oxide copper oxide monocomposite for the degradation of crystal violet dye. My content is introduction, objective, and then gap analysis, methodology, characterization, time frame, and references. So first of all, we will talk about nanotechnology. Nanotechnology is the study and manipulation of matter and incredibly small size, generally between 1 to 100 nanometers. Nanotechnology is the rapidly growing research discipline that investigates nanostructure with a recent focus on metallic nanoparticles. The most extensively studied nanoparticles are silver, gold, titanium, copper, and zinc. What are nanoparticles? So a nanoparticle is a small particle that ranges between 1 to 100 nanometer in size. It is undetectable by human eyes. Nanoparticle can exhibit significantly different physical and chemical properties to their larger material counterparts. The use of nanoparticles spans across a wide variety of industries, from healthcare and cosmetic to environmental preservation and air purification. So we will talk about the copper oxide nanoparticle. Copper oxide nanoparticle is a significant p-type semiconductor a band gap of around 1.2 electron volt. It is frequently used for a large variety of applications like high temperature superconductor, solar cell, gas sensor, catalytic implementation, optical application, hydrogen storage material, and medical application. Owing to the increased surface area and the likelihood of facilitating electron transfer reaction at a lower over potential, they were commonly used for non-enzymatic sensing of clinically important analytes. Copper oxide nanoparticles have shown their ability for pharmaceutical action, especially for anti-tumor therapy. Copper nanoparticles and avoid bacterial infection. Copper oxide nanoparticle can be used as a catalyst for dye degradation, but the photocatalytic efficiency of copper oxide nanoparticle remains still low. By loading copper oxide nanoparticles on graphene oxide, reduced graphene oxide, its advantage can be utilized to obtain higher photocatalytic efficiency. The next one is graphene. So graphene are the layers having two-dimensional structure with high surface area where sp2 hybridized carbon atoms are covalently arranged in hexagonal network. So here we can clearly see that the 2D structure of graphene and fullerene carbon nanotube and graphite 3D structure. Graphene serves as a basic structural unit for other allotropic form of carbon, that is zero-dimensional fullerenes shaped by wrapping up graphene layer to make a sphere, means baki baat. Similarly, carbon nanotube have one dimension are shaped by rolling the graphene sheet to form cylindrical structure, and three-dimensional graphite are formed by piling many sheets of individual graphene layer bonded together by van der Waals bonds. Graphene exhibit extraordinary properties containing such as high exterior capacity, extraordinary inherent flexibility of electron, and admirable thermal conductivity. Graphene and its composite has attracted vast courtesy over the past decade due to numerous applications including membranes, electrode, lithium-ion batteries, supercapacitor, drug delivery sensor, etc. The next one is graphene oxide. Graphene oxide has similar structure to graphene. Both of these compounds have hexagonal carbon lattice, but the graphene oxide sheet is usually distorted where it is bonded to oxygen group. The difference between graphene and graphene oxide structure is here you can see a different functional group includes alcoholic functional group means hydroxyl and carboxyl functional group, and the other one is epoxide functional group. Graphene oxide is one of the source material for large-scale production of graphene has been studied for years. There are many applications of graphene oxide such as potential material for electrode in lithium-ion batteries, transparent conductor in organic photovoltaic cell. Despite of these advanced applications, the fundamental determination of structure of graphene oxide has been slow. The next one is reduced graphene oxide. Graphene oxide is the derivative of graphene, a two-dimensional carbon electrode with single layer of carbon atom arranged in hexagonal lattice. So here we can see that graphene oxide have many oxygen-containing functional group. If we reduce it by using reducing agent, the most of the oxygen-containing group disappears. So here in reduced graphene oxide, there is no carboxyl group and no hydroxyl group. That means it is reduced. Reduced graphene oxide is produced by chemical, thermal, or electrochemical reduction of graphene oxide. This reduction process removes some of some or most of the oxygen containing groups, restoring some of the graphene-like properties such as electrical conductivity and mechanical strength. Reduced graphene oxide exhibits unique property due to its reduced oxygen content, making it promising material in various applications, including electronics, energy storage, sensor, and biomedical devices. Graphene oxide is obtained by oxidizing graphite, introducing various functional groups such as hydroxyl, epoxy, and carboxyl group on its surface. This oxidation process imparts hydrophilicity to graphene oxide, making it dispersible in water and other solvents, which is advantageous for processing. Uh, the another one is crystal violet, which is dye. Crystal violet is also known as basic violet 3, and Argentine violet and methyl violet 10B. So here is the structure of crystal violet dye. You can clearly see that there are 3 phenyl group attached to the carbon, and with the phenyl group there is secondary amine. So crystal violet is a well-known dye and has been used in biological stain, veterinary medicine, additive to poultry feed to inhibit the propagation of mold, intestinal parasite, and fungus, etc. It is also used in textile industry and paper coloration. But the problem is it is mutagen and mitotic poison, so its disposal and removal from the industrial effluent is a major environmental problem. So there are various methods that have been suggested to handle the dye removal from water, like biodegradation, coagulation, adsorption, advanced oxidation process, and photocatalysis. Among these methods, heterogeneous photocatalysts appear to be a promising field of study, which is not only effective for near ambient degradation of organic contaminants, but convert carcinogenic dyes to water, carbon dioxide, and mineral acid. In this project, we will focus on the removal of crystal violet using copper oxide reduced graphene oxide photocatalyst. Uh, the objective of this research is first to synthesize and characterize the copper oxide nanoparticle and reduce graphene oxide copper oxide nanocomposite, and the other one is to assess the photocatalytic degradation potential of repaired nanocomposite using crystal violet dye. Uh, the methodology is first you have to collect a plant and their uh, extract will be prepared, and then synthesis of reduced graphene oxide, copper oxide nanocomposite will be done, and then characterization, and the last one is application to degrade a dye. Reduced graphene oxide can be synthesized by taking 1.5 gram of graphene powder, which will be mixed with 1.5 gram of sodium nitrate and 70 ml of sulfuric acid, and then add 9 gram of KMnO4 in ice bath because it is a very high exothermic reaction. So you can proceed this reaction by using ice bath. The mixture and then the mixture will be warmed to 40 degrees centigrade and stir it for 1 hour, and then addition of 40 ml of water and followed by the addition of 10 ml of 30% hydrogen peroxide. Then the brown color confirm the synthesis of graphene oxide, and then filter and wash with 0.1 molar HCl and water, and then 3 repeated centrifuge at 3000 rpm for 30 minutes, and then graphene oxide powder will be heated at 350 degrees Celsius, will be washed and shade dried, and then Caralluma tuberculata will be powdered using grinder,
and 20 gram of powder will be taken in 100 ml of deionized water and stirred. Filter using Wattsman filter paper and then 10 ml of acetate mixed with 90 ml of 0.1 molar copper sulfate with stirring for 2 hours. The brown precipitate confirmed the formation of copper oxide nanoparticle and then washed with ethanol and dried in 70 degrees centigrade for 8 hours. Synthesis of reduced graphene oxide, copper oxide nanocomposite. 100 ml of reduced graphene oxide suspension will be mixed with copper oxide at different ratios, and it is simulated for 1 hour, and then mounted to reflect condenser and stirred at 95 degree overnight, and then washed with distilled water and ethanol, and then dried. The characterization of the prepared nanoparticle and nanocomposite will be done by using scanning electron microscopy, Fourier transform infrared spectroscopy. In SEM, in scanning electron microscopy, we will deduce the size of the particle, and FTIR spectroscopy, we will check the different functional group, and XRD, we will check the crystallinity of the synthesized nanoparticle, and in UV visible spectroscopy, we will study the formation of copper oxide and its degradation of dyes. So here is the FTIR spectrum of copper oxide nanoparticle, and it is exactly matched with the literature. So you can see different peaks here. Over here is one is 3238 centimeter 2940 2360 1529. Above 1500, below 1500 there is a fingerprint region, and this one is a functional group region, and it is exactly matched with the literature. So here is XRD analysis of copper nanoparticle, and this is same analysis of copper oxide nanoparticle, and XRD of reduced graphene oxide, FTIR of reduced graphene oxide, and this is a UV spectrum of copper oxide nanoparticle, and SEM of reduced graphene oxide. So the application is a photocatalytic degradation of crystal violet. You will take a known amount of catalyst, and it will be dispersed in 40 ml of aqueous solution of 0.1 millimolar organic dye. Drawn and UV visible spectra will be recorded, and then blank experiment will also be performed. Thank you. My presentation is over.
Bismillahirrahmanirrahim. My name is Sabrina Nas. MPhil student in Cohort University of Science and Technology. Here I present my research topic in the International Conference on Biological and Medical Sciences entitled Isolation, Identification and Antibiogram of Bacteria Isolated from Cutaneous Leishmaniasis Wounds in District Karak. Introduction. Cutaneous leishmaniasis is a parasitic infection caused by intracellular flagellated protozoa of genus Leishmania. The disease is transmitted to human via the bite of infected female phlebotomine sandflies. The cutaneous form of leishmaniasis is characterized by distinctive skin lesion and is typically endemic to certain regions. The most common manifestation of cutaneous leishmaniasis is the presence of ulcerated nodules. In Pakistan, 21,000 to 35,000 cases of zoonotic cutaneous leishmaniasis is reported. The cutaneous leishmaniasis has appeared a considerable health issue in characteristic of cohort division. The breakdown of host tissue within leishmaniasis lesion release nutrients such as amino acid and fatty acids that creates an ideal environment for bacterial growth. Wound infection have significantly increased morbidity, mortality, length of hospital stay, and direct and indirect health costs across the globe. If not treated, wounds can get colonized by a wide range of potentially harmful bacteria, making them vulnerable to infection. This multimicrobial illness complicates bacteriological research and is usually disregarded in research constrained environment. When a wound becomes infected, it takes much longer time to heal, which raises the cost of therapy and the degree of pain, discomfort the patient experiences. According to the World Health Organization, drug resistance is hindering the fight against a number of diseases, resulting in the failure of healthcare system and an increase in mortality. The different type of bacteria present in the cutaneous leishmaniasis wounds, which are gram positive and gram negative. This may involve the use of antibiotics to treat the bacterial infection, and these bacteria show resistance to the different antibiotics. The growing resistance by pathogens towards antibiotics has appeared is a big hurdle to public health in the shape of high morbidity and earth rate. Factors like the blend of underdeveloping health symptoms and non-social infection, high rate of infection disease, cheap antibiotics, and rising incomes and increasing the prevalence of resistant microbes. Objectives of this research: the current study is designed to screen the trending antibiotics on secondary bacterial infection of cutaneous leishmaniasis patients and to determine the antibiotic resistance in these bacterial isolate from the patient of district Karak. First objective to isolate and identify bacteria from cutaneous leishmaniasis lesion. Second one, to determine the antibacterial resistance of bacteria in cutaneous leishmaniasis. Approximately 50 sample of infected people will be collected from local area in Karak district. A sterile cotton tip applicator will be used to collect sample and sweeping the surface of the isolated region. Then the swab will placed in sterile container and carried to the Department of Zoology and further process for sample collection. Tests bacteria culture will be carried out in laboratory of Department of Zoology. Here materials and methods: first of all sample collection, second germ staining, third microscopy, bacterial culture, gram staining, biochemical test, antibiotic susceptibility test. Patients of different age group having characteristic of CL lesion on different body parts verified clinically by expert physician dermatologist will be included in this study. The selected patient will be further identified through microscope using James stain microscope. Slides will be taken and then the James staining is applicable on that microscope slides and only the leishmaniasis positive samples will be proceeded for further identification. Here are some picture of my results. First of all, wound swab sample will be taken and the sample will be refreshed and the culture of nutrient broth, bacteria culture on blood agar, bacteria culture on McConaughey agar, bacterial culture on nutrient agar. Then the culture will be identified through biochemical test, that is TSI test, actinase test, and the isolates will be identified, that is we have 7 different type of bacteria will be we have 7 different type of bacteria will be isolated in this research. First is E. coli, second is Klebsiella species, third is Streptococcus aureus, third is Enterobacter species and Pseudomonas species, Serratia species and Shigella species. All species are isolated in different ratio, that is mainly isolated with E. coli and Klebsiella species which present in a predominant rates in the samples. The selected isolate will be further identified through cell morphology, biochemical test, that is gram staining, oxidase test, VP test, and MR test. Then isolate will be tested to the antimicrobial resistance against selected antibiotics, that is penicillin, tetracycline, ceftriaxone, amikacin, vancomycin. The E. coli show resistance against penicillin and tetracycline. Klebsiella species showed resistance against ceftriaxone and tetracycline. Streptococcus aureus showed resistance against penicillin and ceftriaxone. Enterobacter species showed resistance against penicillin, ceftriaxone, and tetracycline. Pseudomonas species showed resistance against amikacin. Serratia species showed resistance against penicillin and levofloxacin. Shigella species showed resistance against vancomycin and ceftriaxone. Most of the isolated species showed resistance against penicillin and ceftriaxone. Conclusion: this study has revealed a wide range of bacteria with an alarming rate of resistance to the commonly used antimicrobial agent. Good hygiene to the use of appropriate disinfectant, monitoring of antibiotics administration and prescription in hospital and drug store be ensured. Additionally, the regular surveillance in the endemic area will help to control wound infection and the emergence of multi-drug resistance pathogen. The prevalence of the leishmaniasis disease will be reduced by raising public knowledge about changing clothing habits and being mindful of insect bites. A large study should be conducted to determine which demographic feature and behavior enhance the chance of the contrasting leishmaniasis. Biological control of sandfly by plantation of Biogynous glabra was shown to decrease the risk of leishmaniasis and its reduced lifespan of sandfly. Use of impregnated bed nets in endemic area which various insecticides like deltamethrin were successfully applied with a significant reduction of CL incidence rate. The cutaneous leishmaniasis prevalence is high in rural area because people have no knowledge about that disease and not proper care of lesion. That's why the leishmaniasis disease increase in the rural area. So people should be tested in previous early the leishmaniasis lesion and start the treatment of leishmaniasis, so we control the leishmaniasis disease all over in the Karak region. Thank you for listening.
Assalamu alaikum everyone. My name is Sabahat Khatak and I completed my research work under the supervision of Dr. Shaukat Shaja, and my second supervisor is Farina Kaul from PCSIR Peshawar. So the title of my research work is the synthesis of magnetic iron oxide nanoparticles for the photocatalytic degradation of methylene blue and methyl violet dye. So first of all, abstract. This work report a clean, green and cost-effective approach for the synthesis of magnetic iron oxide nanoparticle by using Nanorofricnia plant extract. So the nanoparticles were characterized by using different characterization technique that include Fourier transform infrared spectroscopy, energy dispersive X-ray spectroscopy, and X-ray diffraction analysis, and also SEM analysis. So the optical properties of magnetic iron oxide nanoparticle visible spectrophotometer has shown absorbance peak peak at 238 nanometer. Through the SEM analysis, the morphology and size of nanoparticle was shown, while FTIR analysis is used in order to know about the functional group. Here you can say it gives information about the bioactive compound that are present in the plant extract, that is flavonoid, resin, saponins, tannins, terpenoids, carbohydrate, and amino acid that are responsible for the stabilization and the keeping of magnetic iron oxide nanoparticles. Furthermore, the study reports show the magnetic iron oxide nanoparticles. The magnetic iron oxide nanoparticles as used for the photocatalytic degradation of methylene blue and methyl violet dye, and the use of magnetic iron oxide nanoparticles for the photocatalytic degradation gives effective and reproducible results. So the next one is the introduction of the nanoparticles. So what is nanoparticle is, we all know that nanoparticle are the small particle that ranges from 1 to 100 nanometer. So during the production of different textile and leather products, about 10 to 15% of the organic dyes are discharged in the wastewater. So the presence of these dyes in the wastewater have serious effect on human health and environment, because we all know that different kind of dyes is used for the production of different compounds in the industry. When these effluents come from the industry, so it is hazardous for human health as well as for the aquatic life. So the removal of these compounds from the industrial waste is significant environmental issue, because majority of them are toxic and carcinogenic. So the main two dyes that are used in this work, that is the methylene blue and methyl violet dye. So methylene blue is a toxic and thiazine dye. It quickly crosses the cell membrane and accumulate in the parathyroid gland and the central nervous system. And the second one is the methyl violet, that is extensively used cationic and textile dye due to the low cost and high solubility in water. So it is a mutagen. Eye, skin, and digestive tract irritant. Magnetic iron oxide nanoparticles have received much more attention due to the less particle size, high surface area, lower toxicity, biocompatibility, and the cost effectiveness. So in the present work, the synthesized magnetic iron oxide nanoparticles by using nanorophoric leaves extract is used for the photocatalytic degradation of methylene blue and methyl violet dye. So the next one is the objective of my research work. So the objective of my research work is, so the first one is to synthesize and characterize magnetic iron oxide nanoparticle by using nanorophoric leaves extract, and the second one is to assess the potential of synthesized magnetic iron oxide nanoparticles for the degradation of methylene blue and methyl violet. So the next one is about the synthesis of extract, because the plant extract is used for the synthesis of magnetic iron oxide nanoparticles. So to synthesize extract, 10 gram of plant leaves were taken, washed, and ground with a grinder, and was mixed with 100 ml of distilled water. Then the mixture was stirred on a hot plate at 80 degrees centigrade for 30 minutes. The prepared extract was then filtered with a Ward-Manfield paper, and then it was cooled 4 degrees centigrade and stored for further use throughout the research work. So after the preparation of a plant extract, so this plant extract is used for the synthesis of magnetic iron oxide nanoparticles. So the method for the synthesis of magnetic iron oxide nanoparticle is, for the synthesis of magnetic iron oxide nanoparticles, 50 ml of fresh leaf extract will be added to the mixture of 50 ml aqueous solution of ferric chloride and the 50 ml of ferrous sulfate solution. So both these solution is mixed in 1:2 molar ratio with a continuous stirring of 4 hours. So during the reaction, ammonia is adding drop-wise, because ammonia is used as a precipitating agent and the reaction. So after the continuous stirring of 4 hours, the color of solution changes from brown to black. So the black colored solution indicate the synthesis of magnetic iron oxide nanoparticle. So in the present work, the synthesized magnetic iron oxide nanoparticle is used for the photocatalytic degradation. So both of these dye, that is methyl violet and methylene blue, so the optimization is carried out for the both dyes. So here some parameter is shown in the poster presentation. So first of all, the optimization of methylene blue, and after that, that is methyl violet. So first one is the methylene blue concentration effect. So first of all, different solution was made in order to check the lambda maximum of methylene blue, that is 664 nanometer. So when the different concentration of solution was made. So these solution were present outside the lab in the presence of sunlight. So 0.1 milligram of the catalyst is added in order to check the catalytic degradation. So after time interval, the absorbance value is checked by visible spectroscopy. So from the graph, that is the concentration effect of methylene group. From the graph, you can see that the 2 ppm solution is highly degraded as compared to the 10 ppm solution. So the 10 ppm solution is further optimized for the parameters. So the next parameter is the contact time. Yeah, you can say the time effect for the methylene blue. So from the figure, from the graph that is mentioned in the poster, so you can see that when the radiation time increases, the percentage degradation is also increases. So starting from the 0 minutes up to 120 minutes. So it's a 0 minute, the percentage degradation. You can see the dye degradation is quite less as compared to the 120 minutes. So after 120 minutes, the dye acquire a very flat pattern that is shown in the graph. So it is clear and evident that when the radiation time increases, the percentage degradation of the dyes also increases. So the next one is the methyl violet dye. So the parameter for the methyl violet is the concentration effect. So the concentration effect of the methyl violet is given that different concentration solution, different ppm solution were made in order to check the limit maximum, that is the 590 nanometer. So after that, 0.1 milligram of catalyst is added. That is the catalyst in this case is the magnetic iron oxide nanoparticles. So the catalyst is added after that, in the presence of sunlight, the solution was present. So after some time interval, the the color of the dye solution changes from violet to the colorless solution. So the absorbance value, the catalytic degradation is measured by using UV visible spectroscopy. So from the figure, it is clear that the 2 ppm solution is highly degraded as compared to the 12 ppm solution. So the 12 ppm solution is further optimized for different parameter, etc. So the next one is the characterization. So the sim analysis of the synthesized magnetic iron oxide nanoparticle is given here. As you all know, that sim analysis is used in order to check the morphology and the size of nanoparticle. So here you can see the nanoparticle are spherical in shape, and the size of nanoparticle ranges from 1 to 50 nanometer. And after that, the methodology, you can say the methodology of the methylene blue and methyl violet is given at the end of the poster presentation. So the first one is the photocatalytic degradation of methylene blue. So magnetic iron oxide nanoparticle were synthesized to check for their catalytic abilities by adding 0.1 milligram of the catalyst to the methylene blue dye solution. So the sample were withdrawn at a regular interval to analyze the degradation by using UV visible spectrophotometer. The dye degradation was made at 664 nanometer. So the next one is the methyl violet. As you all know, that methyl violet is also called crystal violet. So it is a purple in color. You can say violet in color. So methyl violet dye was degraded by adding 0.1 milligram of the catalyst, that is magnetic iron oxide nanoparticles, to the dye solution. As I said earlier, different parameters like contact time, concentration, and the catalyst dosage effect in the pH are also optimized. The color of solution changes from violet to the colorless solution after degradation. The dye degradation was measured at 590 nanometer as the absorbance value. So the sample were checked at a different time interval by using the UV visible spectrophotometer. And the last one is the conclusion. So magnetic iron oxide nanoparticle were synthesized by using nanodot plant extract, and the methylene blue and methyl violet dye was successfully degraded by using a magnetic iron oxide nanoparticle as a catalyst. So the last one is the references of my research work. So that's all about my presentation. So at last, I would like to acknowledge my supervisor. So, so first of all, I would like to express my sincere and cordial thanks to my respected and dedicated supervisor, Dr. Shaukat Shuja, Associate Professor, Department of Chemistry, Cohort University of Science and Technology, for the pleasant research work. And at last, I would like to thanks for the attention, for all your attention. So thank you so much.
Assalamu alaikum. My name is Shah Rukh Khan, and I'm MPhil scholar from Cohort University of Science and Technology. Today I'm here to present my topic of characterization and antidiabetic activity of Fagonia critica mediated titanium oxide nanoparticles. As you all know, nanotechnology is the emerging field of study, and it has a very, very keen role in many different applications, whether those are connected to medical science or any other sciences. तो जैसे हमें पता है कि डायबिटीज़ जो है ये एक मेजर हेल्थ इशू है आजकल दुनिया में, तो हम नैनो पार्टिकल्स की टेक्नोलॉजी को ही इस्तेमाल करते हुए इसकी क्योर के ऊपर थोड़ा सा कोशिश करेंगे कि इसको थोड़ा सा इन्वेस्टिगेट करेंगे, कोई इसका कोई क्योर या कोई इसका कोई अल्टरनेटिव हमें मिल जाए बेटर। क्योंकि हमें पता है कि जो नैनो पार्टिकल्स हैं ये इनका पता लगाया गया है कि इनकी साइड इफेक्ट्स एलोपैथिक दवाइयों की निस्बत बहुत कम है या ना होने के बराबर हैं। तो जैसे नैनो पार्टिकल्स को इस्तेमाल किया गया है बाकी डिफरेंट हेल्थ इश्यूज को सॉल्व करने के लिए या उसके ऊपर उनके इफेक्ट्स को इन्वेस्टिगेट किया गया है, बिल्कुल इसी तरह हम डायबिटीज़ के ऊपर भी इसको चेक करेंगे। तो इसके लिए मैंने कंसिडर की हैं टाइटेनियम ऑक्साइड नैनो पार्टिकल्स, और इनको हमने डेवलप किया है यूजिंग फेगोनिया क्रिटिका एक्सट्रैक्ट, एक्वियस एक्सट्रैक्ट एक्चुअली। अब हम इस टाइटेनियम ऑक्साइड नैनो पार्टिकल्स को ही हमने इस्तेमाल किया है डायबिटीज के खिलाफ इन वीवो, मतलब हमने इसको अल्बिनो रैट्स के ऊपर यूज किया है, और इसके बाद हमें इसके डिफरेंट रिजल्ट्स मिले जो कि इंशाल्लाह ताला हम आखिर में डिस्कस करेंगे। ऑब्जेक्टिव हमारे स्टडी का ये है कि टू सिंथेसाइज एंड कैरेक्टराइज टाइटेनियम ऑक्साइड नैनोपार्टिकल्स यूजिंग एन एक्वियस एक्सट्रैक्ट ऑफ फेगोनिया क्रिटिका, and the second one is to evaluate the antidiabetic activity of synthesized titanium oxide nanoparticles. प्रिपरेशन ऑफ एक्सट्रैक्ट की बात करें तो हमने सबसे पहले इस एक्सट्रैक्ट को कोहार्ट के लिए एरिया से कलेक्ट किया है प्लांट को, फिर इसको हमने जो है शेड ड्राई किया है फॉर 15 to 20 डेज। उसके बाद हमने इसको पाउडराइज किया, पाउडराइज करने के बाद हमने इसका एक वो बनाया, सोल्यूशन बनाया, एक सस्पेंशन हमने इसका बनाया, फिर इसको हमने हीट किया है 40 डिग्री सेल्सियस पे 30 मिनट्स के लिए। उसको हमने फिल्टर किया यूजिंग वॉटमैन पेपर नंबर 1, और उसका जो फिल्ट्रेट हमारे पास आया तो वो हमारा जो है असल में एक्सट्रैक्ट है फेगोनिया क्रिटिका का, और उसको हमने स्टोर कर दिया था 4 डिग्री सेल्सियस पे फॉर द फर्दर यूज। नेक्स्ट इज सिंथेसिस ऑफ नैनो पार्टिकल्स। हमने नैनो पार्टिकल्स को सिंथेसाइज करने के लिए टाइटेनियम आइसोप्रोपोक्साइड को एज अ सॉल्ट यूज किया है। The conical flask of containing 0.5 molar of titanium isopropoxide and ethanol extract will react under stirring at 40 degrees Celsius to synthesize titanium oxide nanoparticles। 40 डिग्री सेल्सियस इस वजह से क्योंकि हम ग्रीन साइंथेसिस कर रहे हैं, इससे ज़्यादा पे अगर हमने किया तो जो बेसिकली फाइटोकेमिकल्स हैं, उनके जो फंक्शनल ग्रुप्स हैं वो totally denature हो जाएंगे। The synthesized titanium monoxide particles were separated by centrifugation at 5000 RMP for 15 minutes after 4 hours of continuous stirring. The centrifuged nanoparticles were then undergone an ethanol wash, और इसके बाद हम इसको ड्राई किया। आफ्टर दैट कैरेक्टराइजेशन की बात आ जाती है, तो हमने इनको कैरेक्टराइज किया डिफरेंट टेक्निक्स को इस्तेमाल करते हुए, जिनमें से यूवी विजिबल है, फोरियर ट्रांसमिशन इंफ्रारेड स्पेक्ट्रोस्कोपी है, SEM है, XRD है एंड EDX है। यूवी स्पेक्ट्रोस्कोपी को हमने इसलिए यूज किया कि हमें इसके साइज का वो चाहिए था कि इसका साइज क्या है। उसके बाद FTIR को हमने इसलिए यूज किया कि इसके ऊपर जो केपिंग एजेंट के तौर पर फाइटोकेमिकल्स काम करें तो कौन कौन से फंक्शनल ग्रुप्स हैं असल में। इसके बाद जो है SEM को हमने यूज किया। SEM को हमने मॉर्फोलॉजी एंड टोपोग्राफी मालूम करने के लिए यूज किया। इसके बाद बारी आ जाती है XRD की। XRD क्रिस्टलिनिटी के लिए हमने यूज किया कि इसका क्रिस्टलाइन स्ट्रक्चर कैसा है। एंड द लास्ट वन इज EDX, जो हमने एलिमेंटल एनालिसिस के लिए यूज किया कि जिसने टाइटेनियम को कंफर्म किया कि 76% टाइटेनियम इज प्रेजेंट इन नैनो पार्टिकल्स जो हमने प्रिपेयर किए थे। इसके बाद हमारी एसे आ जाती है इन विवो एंटी डायबिटिक एक्टिविटी की एसे। इसमें हमने सबसे पहले माइस लिए। माइस के हमने 3 ग्रुप्स बनाए। एक ग्रुप को हमने कंट्रोल ग्रुप की शक्ल में लिया कि जिसको हमने डायबिटीज नहीं कराई। अगले 2 ग्रुप्स को हमने स्ट्रेप्टोसोटोसिन से ट्रीट किया, तो इंडक्शन ऑफ डायबिटीज मेलाइटिस हो गई थी। अब जब इंडक्शन ऑफ स्ट्रेप्टोसोटोसिन की वजह से जो हो गई है डायबिटीज मेलाइटिस की, तो हमने सबसे पहले उसको चेक किया है कि हुआ भी है कि नहीं। तो हमने BGL टेस्ट किया, ब्लड ग्लूकोज लेवल टेस्ट। वहां से हमें पता चला कि जिन माइस का ब्लड ग्लूकोज लेवल फास्टिंग पे 200 mg/dl है, तो उनको जो है वो कॉज हो चुका था, इंड्यूस हो चुका था उनमें डायबिटीज। तो हमने उनको सेलेक्ट किया फॉर फर्दर स्टडीज। अब इसके बाद अगर हम बात करें उसकी कि हमने इसको ट्रीट कैसे किया, तो हमने अपने नैनो पार्टिकल्स का सोल्यूशन बनाया। ब्रीफली इसके बात करें तो हमने उसका सोल्यूशन बनाया और हमने माइस को 30 mg/dl का डोज दिया एवरी डे फॉर द नेक्स्ट 7 days। और जैसे ही हम डोज देते तो हम 15 मिनट्स के बाद चेक करते BGL, 30 मिनट्स के बाद, 60 मिनट्स के बाद, 90 मिनट्स के बाद, 120 मिनट्स के बाद एंड 180 मिनट्स के बाद। तो हम चेक करते रहते हैं कि क्या फर्क आया है, आया जो ग्लूकोज लेवल है वो ड्रॉप भी हुआ है ब्लड में कि नहीं हुआ। तो जो हमें फील हुआ, जो मुझे पर्सनली फील हुआ, दैट वाज कि 60 मिनट्स एंड 90 मिनट्स के बाद खासा चेंज आया। ग्लूकोज लेवल में काफी हद तक कम हो जाता था और ग्लूकोज लेवल काफी कंट्रोल हो जाता था। जो मेरे एवरेज कैलकुलेशंस हैं उनके मुताबिक जो रिजल्ट्स हैं, दैट इज 82 mg/dl हमारा आ जाता था 200 mg/dl से, जो कि हमारा इनिशियल डेटा भी है। तो यहाँ से ये चीज जो है कंफर्म हो जाती है कि जो टाइटेनियम ऑक्साइड नैनो पार्टिकल्स हैं, स्पेशली जो फेगोनिया क्रिटिका मेडिएटेड थे, क्योंकि फेगोनिया क्रिटिका को मतलब पास्ट सम सेंचुरीज से या सम 1000 ईयर्स से इस्तेमाल किया जा रहा है एज एन एक्वियस एक्सट्रैक्ट फॉर डायबिटीज मेलाइटिस। मतलब इसको क्योर करने के लिए इसको यूज किया जाता था पहले से होम्योपैथिक दवाइयों में। तो उसी को हमने यूज किया टाइटेनियम ऑक्साइड नैनो पार्टिकल्स में, ताकि टाइटेनियम ऑक्साइड नैनो पार्टिकल्स का अपना भी असर हो और जो उसके ऊपर कैपिंग एजेंट के तौर पे फेगोनिया क्रिटिका के जो फाइटोकेमिकल्स हैं वो जो उसके ऊपर आए तो उनका भी अपना एक असर होगा। तो ये एक इनका कम्युलेशन जो होगा, जो कम्युलेटेड असर होगा, तो वो क्या होगा, उसको जब हमने चेक किया तो हमें खासा अच्छा रिजल्ट मिला। और हमें ये भी पता चला कि ये इंतहाई साइड इफेक्ट फ्री हैं। और इसके अलावा बाज माइस में हाइपोग्लाइसीमिया भी कॉज हुआ, लेकिन वो बहुत कम केसेस में ऐसे हुआ क्योंकि वो हमसे ज़रा डोज ज़्यादा हो गया था। तो ओवरऑल अगर हम इस स्टडी को सम अप करते हैं तो जो टाइटेनियम ऑक्साइड नैनो पार्टिकल्स जो हमने प्रिपेयर किए उसका जो असर है वो एक इंतहाई बेहतरीन किस्म का असर आया। और जो हम एक्सपेक्ट कर रहे थे इसकी एफिशिएंसी, तो इसकी एफिशिएंसी है। इंशाल्लाह ताला होप फॉर द बेस्ट के फ्यूचर में इसको न्यू ड्रग डिजाइनिंग के लिए इस्तेमाल किया जाए। थैंक यू।